elevating Indian healthcare to global genomic standards, redefining what's possible, where innovation meets safety.
Morning, everyone. It's kind of custom to give up at least 30 minutes for the god of traffic in Bangalore. So apologies for the delay, but nonetheless, let's start. Before we begin, I would request you all to keep your bags down because we will love to capture your beautiful faces than the bag handles. Anyway, nonetheless, let's start now. Welcome to BioVerse 2026, the 10-year user conference of Azooka Labs. 10 years is a very long time in science and technology, especially in genomics. And what makes today special isn't just the milestone, it is the ecosystems that help build it. We're truly delighted to host such a distinguished gathering of clinicians, technicians, innovators, investors, and ecosystem partners. From genomics to microbiome research, Azooka has spent a decade building from India for the world. We have an exciting lineup ahead, conversations that sit at the intersection of genomics, AI, diagnostics, and real world healthcare. So let's begin. I'd like to invite Alex to formally open BioVerse 2026, our co-founder.
Hello. Uh, Good morning and welcome to BioVerse. So my name is Alex T. Paul. I am the co-founder and CEO of Azooka Labs. I happen to successfully survive among the scientists for 10 years without being a scientist. That itself I'll consider as an achievement here. So I'll kind of introduce us what we do here. So I'm here to share some basic small talk before things take off. Uh, just a minute. Yeah. So before actually starting this Azooka, I was with a software company, happily doing stuff. Then I had a moment of truth where I believed that I should work on something that really impact lives. That's when my friend and co-founder, Dr. Fatima, got in touch with me with her PhD thesis, saying that she got something really interesting that the world needs to know about or the world needs to benefit from. That's how we started the journey with a product called as Tinto Rung, India's first safe DNA binding molecule. Luckily, I didn't have to make a presentation at all because the first investor happened to be my college friend Vardarajan, who actually parted with money without even me having to make a pitch. I told him, I'm doing something, I need money, transfer it. That's it. This was my pitch, and he transferred the money without asking any questions. So the presentation is rough on the edges because it's all a 10-year memory. So kindly bear with it. So the first office that we got from SID, I have to say this, we failed in SID for close to FSID, it is as it is known today. We failed in FSID for close to 1 year, I think we met with FSID close to 5-6 times. We failed around 5 times, and that failure actually shaped us to come back with more and more convincing presentations. And that's when we got into FSID, and that itself helped us to win advisors who saw our resolve and decided to help us with the rest of the journey. So this is our first office that we got. It was less than 200 square feet of space. And with that, we thought that, oh, we've done something important. We are going to go to the market and kind of capture the market without understanding the chemistry behind it. So we actually set up our first unit, our first manufacturing unit in Koramangala, hired a team, went to market. This is our first box and brochure that was done 10 years back, kind of saying that we are going to launch an eco-friendly product. People are going to line up and buy, and we're going to really capture the market. And this is our first MVP, Tintorang DNA and RNA stain that was launched. And we did a major launch with the health minister, who came and launched our products and gave us that oomph factor. And we went out and gave free samples to customers. It was in our car, we packed lunch, and we used to visit customers all over to kind of deliver samples. And and guess what? Okay, the market was not ready for it. Okay, I'm sorry, and guess what? The market was not ready for it. Okay, we had zero customers, and customers said that, I would really prefer a biohazardous product because it is saving me costs. Okay, so that was and the DNA and RNA based research was not really picking up big in India. And then COVID hit, and with COVID hit, what happened was it was ISC had a pandemic protocol which allowed only scientists to get in. I was not even able to get inside ISC to pick up the stuff out of the lab. It was a bad scene. And then what happened was WHO came up with a mandate that all diagnostic tests must move to PCR and RT-PCR. Okay, and all of them had to upgrade to molecular-based sample collection. And that came as an opportunity for us. At the time when the world was suffering a crisis, I think I have to thank the incubators from FSID, because we almost gave up, because we had great ideas, but we didn't have manufacturing space. Okay, so what will you do? So that's when FSID's one of the startups had come up for sale, and I was asked to go and negotiate a deal with In Vitro Sense. And guess what? Azooka had zero money in the bank, and I myself didn't have any money to, go and make the deal. But we actually negotiated on an empty pocket. Okay, but luckily the deal went through because the financier was FSID. Okay, and we made our first acquisition. Apart from this acquisition, what happened was the lab came with certifications already. The lab came with people. We didn't have to hire people. We didn't have to get the certifications. Even the boxes, materials from the sellotape was already there. We just had to get in and start manufacturing. And we did manufacture India's first biosample collection kit that sold around 1 million units during COVID. That gave us the resolve that we could do what is possible with the most nimblest of teams. Okay, in products, important thing to understand is it's not how big your team is. Okay, it's how effective your team is. What is the nimblest team? With the smallest team, we scaled up to around 50,000 units per day. That gave us the resolve that we could actually build products at scale, and that was RNA wrapper. And the important thing is there are three main incubators who helped us in this process. It was FSID, Ramaya, Evolute, and IKP. So it brings us to the topic. I'm just going to go back to one slide that I kind of missed in the first part, which is this. Okay, friends, team, mentors, investors, partners, and customers. Okay, so this is all This is all a segment that you start with, okay? But actually, the lines are blurry. Friends can become investors, okay? Friends can become mentors, team can become family, mentors can become friends, investors can become friends, partners, customers, all of them, the line is actually quite blurry. So what we found was, we found friends and investors, we found friends and customers who actually adopted us, gave us the support that we needed to reach where we are supposed to be here. So this is some one thing that I missed here is the customers here. Okay, so the really sorry about this. Yeah, so my investors were uh, all ex-Zoho friends who worked with me. So most of the time, I didn't know these people. Okay, most of them happened to be when I was working there. Arun was my designer. Arun is the CEO of a company called Silai.com. Balaji is the vice president of Accenture in the US. Vinod Jain, who's our first investor, is Vinod Jain here? Okay. Vinod Jain is actually a garment wholesaler. Okay. He's got nothing to do with molecular biology or biotech or anything like that. He was just fascinated with what we are doing and came up and invested the money, believe me, without a pitch. He just saw our products, he just saw our execution and said, whatever you're doing, I want a stake in that. So, so that was interesting part. The best part of most of the time we got money was without a pitch. And this is what we ended up building is the moat, is a moat that we got a global patent in 4 countries. And after that, we developed. So, what we did was we developed the DNA binding molecule, and because it was supposed to be license heavy, it was supposed to be licensed. We didn't manufacture the molecule because it was very expensive. We only did it for licensing. But we understood that India was not able to adopt it because step 1 and step 2 was missing. So we started manufacturing the sample collection kits and the extraction kits. That became a hit, and that is covered by trade secrets. I'm just going to leave some of the stuff for Fatima to talk about some of the coolest stuff in the microbiome space that we are doing. But we came up with 54 plus products, and and we have around 200 customers who are using it on a daily basis. 400 customers at the moment, split between both diagnostic labs and molecular biology. So, so this is our manufacturing unit in Pina now, that is serving up to 400 customers. And what we have done is we have built India's first company with the widest range of sample collection for genomics and microbiome, uh, rivaling with Strec Tubes, which is a $75 million company, another company called Zymo, which is $30 million. We compete with Thermo Fisher, we compete with Paxgene. All our competitors are in the US. We are the only ones trying to fight from India and replace all our competition. So, where are we headed next? So, we are headed to a unified health stack where we believe that the health stack is going to be today. What we are seeing is snapshots of health where you take a blood test that is valid for 2 weeks, where you take any kind of test that is only valid for around 2 to 3 weeks. What we believe as the future of health is a stack where we start with the DNA test overlaid with gut microbiome, right on top of the gut micro, which is we call as the functional data, and then we overlay that with the blood and hormone biomarkers, and wearable data comes on top. This is what is the minimalistic. I know that you could overlay proteomics or metabolomics, but it just complicates the layer. This we believe is the most minimal upgrade or most minimal health stack that needs to be in Apple Health or that needs to be in Android Health, which is the app available on your phones. We are working with NetApp for a possible presentation to Android, Google's division on Android Health, to see today your Android Health doesn't store bioinformatics data such as DNA data or gut microbiome data. We have been working with them to see if we can introduce this by default in your phones so that you are pushed more to take the test and make sure that you have a complete coverage of your health. That is what the future remains. Off to, so this is a quick view of what we have today. This is my small talk here, and we are off to the next presentations, off to a great start. I'm not going, I mean, eventually you're going to find out from our customers and partners how our products are being used by them, how we are trying to get into a segment of, we started with diagnostics, but what we see a lot of traction is we've kind of moved into the health, wellness, beauty, and the cosmetics game that has a lot of repeat customers and easier penetration in a lot of markets because people don't want to take a diagnostic test. Everybody wants to take a skin test. Okay, I used to go to places and say that I had a tough time when I went to when I went back to Chennai to my people and say they asked me, Alex, what do you do? Okay, I used to tell them I'm working in genomics and everything, and I usually get an understanding nod or sort of understanding nod saying that okay, he's trying to something. The minute I say a skin test is when I hear from people saying that, hey, can I take a skin test? Is what people say. So I believe that skin or beauty is the easiest game for people to understand. And I think there is a small QR code for you to try your skin analysis. We are trying an app where you can actually do a skin analysis and get reports so that you'll be able to understand the efficacy of the beauty products that you're trying. Okay, so with that, I'll close. If you have any questions, I'll be around to take that later. Off to a great start. Have a great BioVerse days. Thank you.
Thank you, Alex. Before we call upon our advisors, we would like to felicitate them first. So I'd like to call Dr. Benna to felicitate Professor Savitri. Professor Savitri.
Yeah, I'm indeed very delighted to be here for the BioVerse 2026. As Alex mentioned, 10 years is a long time, but their journey has been very, very full of ups and downs, and they have survived. That itself is a point at which worth celebrating in that sense. So I do wish Azooka all the best, and in the next 10 years I see a exponential increase in their product sale as well as research activities. I hope they'll continue with their innovating research and product development. All the very best to Fatima, Alex, and all the members of the Azooka group, and all the best.
I would like to call upon Alex to felicitate Dr. C.V. Natraj. Now Shika Singh to felicitate MRN Muthi sir.
I don't think I deserve any felicitation. It's very kind of Azooka to felicitate me. All that I can say is that Fatima was in my lab as a postdoctoral person, and when she wanted to start her own project, I just said, yes, go ahead. Other than that, my contribution to this company has been absolute zero. But I do appreciate the gesture they have shown, and I wish Azooka all the success in the future.
I think Professor Murthy is just being very humble here. It was him, he opened the door for me and it changed my entire life. I still remember he did ask me to bring Alex to meet him. He did a 1-hour interview for Alex, after which he said, okay, I think you can do it. It may be worth trying. And I was in the Dr. D.S. Kothari postdoctoral fellowship. I was getting that fellowship for a research I had to do at his lab. And he said, I'm sure Kothari is not going to be upset if you're going to employ 10, 20 people, so it's okay, you can do your work. So he's just being very humble here. But I think it was him and his lab who opened the door, even for the postdoc. I don't have a background in structural biology. I come from a plant biotechnology background. So, but when I had applied to him, he said, just for your passion and your interest, I'll open my lab to you. So that changed my life, and I think I definitely owe it to him. Thank you, sir.
I now call on Siddhi to felicitate Dr. Usha.
Really happy to be here. Congratulations, Fatima and her team. All the best. I hope you continue to do well.
And finally, Jose James to felicitate Sridhar Ramanathan sir.
Benazir, you know, the problem is few words and me don't go together. So, which is why I safely avoided. But again, thank you, Alex and Benazir. Congratulations to you and the whole team. 10 years is incredible. Having been part of startups myself, it's truly memorable. Of course, I remember the time that you guys called me over phone before my IKP days to discuss about this new product, Tentorang, and I was going gaga. But I think the biggest lesson for any entrepreneur is you guys and your positivity and seizing the moment, pivoting when you had to, and never ever losing this smile. I've never seen Alex, at least Bena may have some signs this way, that way, but Alex never loses that smile. Keep it up, guys. Congratulations.
We now move on to our keynote address. Our keynote speaker today truly represents the intersection of science and industry. Often described as a chemistry wizard, Dr. C.V. Natraj earned his PhD in chemistry from the Indian Institute of Science at the remarkable age of just 22, setting the tone for a career defined in scientific depth and precision. With a postdoctoral research experience in biochemistry from the University of Michigan, He went on to lead the research as director on the board of Hindustan Lever Limited and later headed corporate research globally for Unilever as senior vice president. Today, he serves on the board of Tata Chemicals and is also a technical advisor to the Society for Innovation and Development at IISc, continuing to bridge academia, innovation, and industry at a scale. And when he isn't driving a large scale scientific innovation, I'm told he's just as precise on the golf course. So please join me in welcoming Dr. C.V. Natraj.
Thank you very much for that very kind introduction. It is certainly a privilege to be here today, Alex and Fatima. 10 years, it doesn't feel like 10 years, it feels a lot shorter than that, but clearly it has been that long. And from what I can see, on the basis of the short presentation that Alex made, it's not diverse by accident in terms of where you are. There is a certain set of events that happened, and I had actually recognized what COVID did along the way. And the fact that it's an evolution and not an accident that you have this level of diversity in what you're doing. But what I will say in the next few minutes is a bit more about technology and industry. And for this, one has to go back to the time of independence, in a way. As you all know, when India became independent, the British had managed to loot the coffers completely and leave the country more or less penniless. And in this state, the leadership of the country at that time had to make a decision in terms of how they were going to set up the various industries that was needed for India at that time, starting from making energy, for instance, energy generation, steel making, metals, all the way to consumer products like sabun, and on the first floor today you have Karnataka soaps and detergents, which is just a coincidence. Again, the manufacture of detergents, including sabun, was all part of something that it was felt that the capital needed to start any of these industries was entirely in the hands of the government. And therefore, the so-called public sector undertakings made a beginning at the time and have remained a very significant part of the manufacturing activity in this country ever since. One may wonder, life is like a one-way street. It's difficult to imagine what other options existed and what else could have been done other than raising money or capital from the government. Are there really genuine options available? And I'll just take the example of South Korea, because that's always been an interesting example to take. South Korea became independent a year after India did from Japan. And if you think about a metric to compare the two countries, the best metric that I can think of is per capita GDP, which at that time was about the same for India and South Korea, give or take $350. Fast forward to today, the GDP per capita in India is in the suburbs of about $3,500. That's about a 10-fold increase in the GDP per capita for India. I don't think that many people would have any surprises if I told you that the South Korean GDP per capita is one of the higher ones in the world, is about $40,000, 10 times larger than the GDP per capita for India. The path that South Korea chose was to privatize industry from day one. There's very little public sector investment in manufacturing in South Korea. And I need hardly tell you that 4 or 5 globally recognized multinational corporations are based in South Korea. We don't have any. So it is worth examining and trying to understand a few lessons in terms of perhaps what could have been. The public sector undertakings, the way they were structured from the very beginning, was that there was a certain method of starting the business, and that was called what became known as the license start. So you needed a license to manufacture anything in the country at that time. So without a license, you couldn't set up any manufacturing. The license came with a cap on the capacity that you could make. Suppose you got a license to manufacture cars or scooters, it would specify how many cars or how many scooters you could make, not one more. And I can tell you that as part of the industry at the time, we had a certain licensed capacity for the manufacture of personal care items, toothpastes. And one of the key decisions we used to take around September of every year, or August, September of every year, was when to stop production. Because if you exceeded your licensed capacity, the punishment was draconian. So you had to be very careful in managing this. What does that mean? So when you cap production, and because the demand is massive, the supply is limited, there is zero incentive for innovation. So one of the key things that happened as a consequence of the licensed capacity and a cap on the capacity was the disincentive for innovation of any kind. When you talk about innovation in industry, the key driver for innovation is the consumer. When the consumer has an option of more of where the supply at least meets the demand or even exceeds it, they have an option to choose product A or product B. Then you need to be ahead of your competition to be able to do that. And that is where the innovation in industry basically comes from. Now, this was not as, this sounds like a very depressing story, but there were exceptions. And one of the biggest exceptions that I can think of at the time was the pharmaceutical sector. The pharmaceutical sector went on to innovate in a big way the generic molecules, but it was not just the innovation of the molecules, but it was also about the formulations, the markets, getting the licenses, getting the approvals, and getting everything ready to go to market the day the patents ran out. The fact that they have been incredibly successful is that they are the backbone of very health systems, like the NHS in the UK would be more or less on its knees had they not had the access to the generics that come out of India. So there have been exceptions to this. It's not as though everybody sat back and lay down. Some people did do. The bulk of the industry in India did not innovate. If you come, and at the end of 90s or in the beginning of the 90s, when the country was forced to liberalize on the back of a series of events that happened, almost all of these industries which had stayed well clear of innovation for all this time perished. You did not have them within 5 years. Most of these industries which had existed on the basis of their access to a license no longer existed. But one of the key things that I have seen, and I was expecting in the 90s that industries would now come running to academics to find solutions to problems, that did not happen at the rate at which I was expecting it to happen. There are a whole range of reasons, because many of the industries at that time felt it was better or more convenient to access technologies, however substandard the technologies were, from anywhere in the world, and have the technology transferred done, rather than actually work on developing their own technologies. One of the mistakes in bringing in such technologies into your own industry in this country that I have found is that there are some a very key point that becomes a difficulty is the India-centric nature of the solutions. And I'll come back to this in a minute, but one of the best examples I can think of is what Alex was mentioning earlier as an example. What is an India-centric solution? An India-centric solution, for instance, for me is that when you have a diagnostic to be done anywhere in the world, you assume a certain integrity of the cold chain. In other words, you collect the sample, you drop it into ice, it goes into a cold chain, The transportation is cold, it arrives cold at the destination and goes into the refrigerator and is subsequently analyzed. That is a cold chain. Not now, but when we used to have an ice cream industry in business in Hindustan Lever, one of the biggest challenges we faced was the integrity of the cold chain. We could not supply ice cream anywhere other than in the major cities and towns. Our rural distribution was practically zero because we could not supply an ice cream that had gone through a non-integrous cold chain. If it has melted and then refrozen, that was not ice cream for us, and we wouldn't sell those ice creams. So for a long time, I have recognized that the integrity of the cold chain in this country, and even now, and it will continue to be so, because we have struggles with power supply and a whole range of things. But so therefore, a solution to this collection of samples, retaining the integrity of the samples at the time of evaluation, which may be a day or 2 days later, is a specific India-centric problem. And a solution for that is the kind of technology that we are talking about. When you say India-centric problems, India-centric solutions, those are the kind of things. And what Azooka has done is precisely that. It is a very specific problem, which is very local, but it has a number of applications that will follow from here, which are actually very, very relevant. But coming back to where I was ending up, the key about the interaction between academia and industry begins first and foremost with the identification of those problems for which relevant scientific solutions can be found. And this dialogue, unfortunately, does not happen very well with large established industries and academia, because the language needs a little bit of an interpretation. And this is where, and I very strongly believe, that the role that startups play in India in building technology into industry at all levels is very unique and very specific. And the reason why startups make this distinction is that entrepreneurs have a way of taking a problem that has a number of, virtually insurmountable problems built into it, put a lot of passion and energy and creativity and solve them and reduce those to a proof of principle or a proof of concept idea. Any conversation with anybody, especially let's say you're a major industry and you're an academic, that conversation is like a railway track, parallel lines. Neither understands what the other is saying. But the moment you put a proof of principle or a concept or a product on the table, that conversation is now brokered. The dialogue is now possible. And that is the role that startups play going forward. And it has been the case for about 20 years. And what is really interesting is that in a place like India, especially India in Bangalore, the number of academics from Indian Institute of Science who have incubated companies with us is about nearly 40% of the 150 startups that we have come out of academics in Indian Institute of Science. So this, according to me, is one of the more important movements that is happening, where academics are now beginning to see their way into embedding their discoveries and science and technology into industry through the startup. The dialogue happens through the startup, but the end result will be in the fullness of time. The scaling of that will actually happen in industry. So although I started on a somewhat negative note at the beginning, I think the future is a far more rosy and exciting possibilities for embedding technology in our industries. I will stop there because I'm sure I have overshot my time, but it is once again a great pleasure to be associated with Azooka on this occasion. A very interesting 10 years, but hopefully a far more productive, scalable, and enjoyable 10 years in the future. Thank you very much.
Thank you, sir. That was both thought-provoking and grounding. I would like to call upon Kumar Shankaran to felicitate the keynote speaker. We now move on to our next speaker, where both brain health and AI move into a very fascinating space. Lina Emanuel is the founder and CEO of Brainsight AI, where she is building AI and 3D visualization based on solutions to transform how neuropsychiatric disorders are diagnosed and understood. With over 2 decades of experience spanning technology, public health, and business, Lina is driven by one constant: building systems that create meaningful ethical change. She began as a systems programmer at NetApp and the Offshore Development Center at Kisco at Infosys, moved into international development and health policy, and later deepened her focus on healthcare at the Indian School of Business. Today, she brings together technology, policy insight, and entrepreneurial conviction to reimagine brain health. Please welcome her.
Hi, everybody. Thank you so much for having me here. It is such an august gathering, and to be able to speak after the people who actually shaped my childhood is an honor. And I shouldn't have been here in the first, but like I have a flight to catch, so I need to leave early. So I will be talking about our work at Brainsight AI. Okay. Let me start with talking about the brain is actually one of the most, sorry, how does this work? Okay, cool. All right. So the brain is the most complex thing on earth, and it's in a lot of ways the next frontier for a lot of people to work on. Now, in the past decade, an area of neuroscience called connectomics, which is looking at functional connectivity of the brain, constrained by structural connectivity, has become robust. Now, and the reason why it has become robust is because over the past decade, scientists have been working to map the connectomes of C. elegans, which took 16 years, melanogaster, which took 12 years, and now have been looking at how to map the connectome of the human brain. Now, there are different ways to be able to do this. There's the connectome, which is, Google has given one of their largest grants to the University of Zurich, where they look at mapping structural connectomics by cutting the brain into tiny slices and using that to be able to map the structural connectivity. We use MRI of a person to be able to map the connectome. We use 3 types of MRIs, one called the structural MRI, the functional resting state functional MRI, and the diffusion MRI. These are 3 types which look at water diffusion, oxygenation, and the structure of the brain. So you can actually see, you think of it like the Google map, so you can actually see the structure, the road, and the traffic of the brain. And when you put these 3 things together is when you're able to, that's known as a connectomics. And one of the uses of this is to be able to map the functions of the brain. And so what we are able to look at are things like your language function, sensory motor function. Those are your more traditional functions of the brain. But apart from the more traditional functions, you can also look at cognitive functions of the brain. So you can look at things like the attention network, the default mode network, salience network. These are networks which are more difficult to map and are new in the space of neuroscience. So the fact that you're paying attention to me is because there's an attention network in your brain which is getting activated. The fact that there's a salience network which is allowing you to block out other distractions and then come into and then be able to pay attention. These are some of the things that we are able to map. When we started out, and I loved what Alex said, I am a software engineer who got into biotech by mistake. When we started out, my co-founder is a PhD from NIMHANS. So she was working on the use of connectomics for psychiatric disorders like bipolar, schizophrenia, major depression, and how connectomics can be used for that. I met her in 2019 and we came together, and I had come from a public health background. I, in fact, sir, used to work in the cold chain for the vaccines development chain in the National Institute of Health and Family Welfare. And the problem of the cold chain is actually quite big even now. So there's a lot to be done there as well. But I came from public health and software engineering background, and then I met her. And I knew that mental health was a really huge problem, right? It's one of the most biggest global health problems. And here was her work in connectomics, which was one of the most cutting edge. She had a paper, their lab from NIMHANS had a paper in Nature Schizophrenia where they showed that connectivity patterns is probably the only body-based marker for psychiatric disorders. And so we decided that we will come together and build a company. And I think we did not know that we were only one of two worldwide companies doing this. We thought that, okay, this is an interesting problem, let's try and figure out. With zero experience in building a company, with zero experience in healthcare, with zero experience in trying to bring an advanced technology, I believed our ignorance was key to being able to build this, because if we were more smarter, we would have probably taken up other problems. So today we are one of two companies, one of three companies. There's now a new one which has come out of the University of Washington. And we are, out of those, we are the only two leveraging AI. So connectomics is really hard. Like there's 4 billion neurons talking to each other. And so you need to be able to map that. And so if your computational power is not enough, you won't be able to map it. And what AI does is helps to remove a lot of those constraints by breaking up the problem space. So we are one of two worldwide working on that. Now we are known, recognized, as I said, we started with zero ignorance, and we are now recognized in India as well as internationally. We were known as the top AI 100, the top 150 digital health companies. So I'm very excited about that. But this is a very quick view of what we build. So as you can see, these are three different types of MRI. One is your structure, which is very clear. It looks at the structure of the brain. The second is your diffusion MRI, which looks at water flow in the brain. And the third is the resting state functional MRI, which looks at oxygenation in the brain. And we use a number of different computational neuroscience algorithms plus AI to be able to map what you see here, which is the tracks and the networks. Sorry, this is not moving ahead. I actually want to talk a little bit about what connectomics allows for. So today we are in over 100 hospitals. 52 of them are commercially working with us, and the rest are in the stage of piloting with us or doing clinical studies with us, where they use it for brain tumor surgery. So what they do is they use our tools to be able to map the functions of the brain before a surgery so that they can ensure that a patient gets out of the surgery with zero functional loss. And we've had stellar results across all the hospitals that we've worked in, where they have seen the value. And we keep getting a lot of their images from the hospital where they say that the patient has come out with zero functional loss, and it makes us really happy. On a side note, I think I don't come, as I said, from a biotech background. And so in the beginning, when I started this company, doctors used to send us open brain pictures, and I was like, oh my God, what is happening in my life? So anyway, so that's on the functional surgeries part. But apart from that, your connectomics is also one of the earliest indicators for dementia, much earlier than eye scans, much earlier than virtual reality solutions, which look at dementia around 3 to 4 years. Your connectivity patterns in the brain actually are able to look at it around year zero. Similarly, my co-founder's PhD was in the fact that it's the only body-based marker for psychiatric disorders. And what it can really help is in identifying how to do neurostimulation. So even at NIMHANS, even outside India right now, at-home neurostimulation is a very big solution for patients with major depression, OCD. So to know where to place the patch is a very big problem. And so that being able to map this connectivity patterns can help your patients with that. Sorry. I'll talk a little bit about our team. Our team is now a 50-member team. I'm very proud of the fact it's 28 women and 22 men, and it's like it's a very well-balanced team, which is working out of JP Nagar. And we have been working with, let me actually show you, I won't go into all of this, I'll actually just show you all the hospitals which we work with. I'm sorry, this is really slow. This is, I'll just leave with these set of things. So what you can see is all of the recommendations that we've got from hospitals across the country where they have used our work for functional analysis. You've seen, we work in the West, we work in South, and we work in North, and you've got all of them here. We now have done over 2,000 live surgery cases. Of doctors who work with us. So, oh wow. Okay, I think I'll just take a moment and show you that, and with that I will end my conversation so that you have a sense of. Oh yeah, okay, that'll be great. Yes. Yes, so this is all of the doctors. I recently actually sat down and put the pictures of all of the doctors, and this is one of my proudest slides because, hey, these are all of the doctors across the country who are using our work for their surgeries today. Thanks for giving me an opportunity to talk about our work. Happy to take it from here and happy to answer any questions that you might have. Thank you.
The floor is now open for Q&A if there are any questions.
What next?
Yes, so we've actually started working in epilepsy right now. So in fact, I was yesterday at Yesterday I was at Aster where I met patients with epilepsy who've had a very, very unfortunately torturous life when they could have had a really good life. There's so much stigma associated with epilepsy. So we are starting to now work on epileptic foci. How do you map the epileptic foci of the brain so that surgeons can take out only that one tiny part and the patient can go back to extremely good results in the next few, immediately. We are also starting work on stroke rehabilitation where they can use it with RSF MRI. You can actually reduce the time for stroke rehabilitation from 6 months to 3 months. And that's the kind of work that we are now looking at working with various hospitals. So there are lots of things that we can do with our work, but we have to go very slowly. So anyway, thank you. Yeah. Yes, ma'am.
Very creditable that you started in 2019,
Yes.
and now in a matter of 6-7 years, you have so many clinicians who have come forward to use the technology, and that shows that the clinicians are also getting very, very, updated with the technologies that are available and moving forward with that. So it's very, very impressive, and it was nice that you took this bold decision, and also that you have a very big group already joined up together, and wish you all the best.
Thank
Very
you, ma'am.
impressive.
Thank you, ma'am. I really appreciate it. Thank you. Great. Thank you so much. Thank you. Thank you.
That was mind opening in the literal sense. So, thank you so much, Lina. I would like to call upon Professor Savitri to felicitate our speaker. We will now move on to our next speaker. Kumar Sankaran is the founder and the CEO of Lucy Enriched Bio, a pioneer in microbiome research that is reshaping the health and wellness and fitness space across India and Southeast Asia. As a biotechnologist with over 2 decades of experience in systems biology, he previously co-founded Omics Solutions India and has worked extensively in building scalable biological data platforms. Under his leadership, LRB launched BugSpeaks, India's first gut microbiome test, bringing personalized gut health into our mainstream. Over to you, sir.
Good morning, everyone. First of all, Alex, Fatima, thank you for inviting me to this event. All the Azooka. I spent 30 minutes in front of my wardrobe this morning, trying to figure out what to wear. I'm usually dressed up in a suit like this, but it was Azooka's event. So I wanted to, try and be as cool as Alex is, wear a t-shirt and wear a jacket. I tried, but, I can't pull off that swag. So decided to go in the safe route. My journey has over the last 11 years has been delving deeper and deeper into the microbiome world. And we started the company in 2014. Back then hardly anybody knew about the microbiome, and before starting I thought it was pseudoscience, and now become a firm believer of it. So what we would like to do with microbiome, the first few years of when we were doing a lot of R&D, we were generating a lot of the microbiome data. I see Shivanjan in the audience, who's been very helpful during those early days, who was trying to get us clients who would just like to generate microbiome data and analyze a little bit on it. From there, we moved into actions, and now we've come to a stage where we can actually start looking at outcomes of the microbiome. I'm sure you've seen various versions of the slide, what the microbiome is, and especially the gut microbiome, and the kind of microorganisms that are there. But what is most important to us is it is actionable. By food, by medication, by lifestyle, by geography, all of that changes the microbiome. And that is what makes it very, very interesting, both from a disease aspect as well as from a health and longevity aspect. From all of this, it is actionable, and hence makes it very interesting. we focus a lot on chronic diseases, and that's this is the epidemic that is happening globally. Whether it is the metabolic syndrome from diabetes to obesity to the gut-brain axis with Alzheimer's and Parkinson's becoming common as we age. But what we're trying to do is bridge that data gap. The reason I say this is I think all the world's knowledge combined, we probably understand less than 10% of the gut microbiome, let alone all the other microbiomes around us. So there is a constant endeavor to make this gap narrower and narrower, which is why it is important to measure reliably. I mean, we might not understand 90% of the data that's there, but if we can reliably measure it and remove any doubt whether the data was measured properly or not, that is the first key. From there, if we can clearly explain what is happening and then be consistent about it, it helps a lot of the healthcare professionals take their decisions further. Using this principle, we've now analyzed more than 2 lakh Indian microbiome samples from all parts of India, and I think we've covered close to about maybe 4,000, no, maybe about 2,500 to about 3,000 pin codes within India. So if you ask me what my job in office is, it's making sure that I talk to Blue Dart and get poop samples from all over the country. The journey has been quite interesting. We won the Frost & Sullivan Award in 2019, which was the first recognition that we got for microbiome diagnostic. And in 2021, we got the National Startup Award for the best life science company that year. The key circle came in 22, 23 when we were partnering with Azooka, where we were expanding globally, and we got a credible Indian technology. I'll talk about it more in later, where we could go globally with all confidence. Sorry, some of the words are missing in my slide, but we're trying to build the microbiome revolution in three shifts. One, by not looking it as a single organ, but as looking at an overall system. The bacteria doesn't know that this is gut or lung or blood. I mean, so we're looking at it at a holistic system. Secondly, we're moving away from generic therapies to personalized therapies. For example, there's what we've started and pioneering in India is looking into personalized probiotics. I mean, instead of taking over-the-counter probiotics, not knowing what is already there and what is missing, can we, based on the report, come up with a personalized concoction for a person? And that's been going, I mean, we've been getting incredible results on that. And lastly, I mean, there is, in healthcare, we always see that most of the reports are tailored towards the healthcare professionals. They're the ones who understand the results and kind of interpret it to the consumer. One thing that we've been trying to do is build a little more consumer education in the microbiome field. It's complex. Most of the stuff is very, very hard to explain, but we are trying that. I mean, this is a constant endeavor to make it simpler and simpler. The way we do this is collect the sample, which is a very, very important part. There's the way the sample is collected and how it's stored and how it's transported to labs. Once it comes back to our lab, we do shotgun metagenomic sequencing, which ensures that we get all kinds of microorganisms, not only bacteria, but we look at fungus, virus, and all the other microorganisms that are there. And lastly, doing the mapping and the interpretation. I think our core IP, I'm hoping our patents will be granted this year, is around the analytics part, where we're looking into interpreting all of the data that is coming out of it. So interpretation of what we're trying to do is the key. In our experience, we kind of learned it the hard way, that the best report is the one where the person reading the report can make a decision within the next minute. He understands, or he or she understands what is happening in the body, and be able to make some kind of decisions based on that. Either it's the consumer itself or the healthcare professional. So that is what we are constantly looking at. And which is why we're going through iterations of our report, where we're trying to come up with scores. Unfortunately, in the microbiome world, there is nothing standardized. I mean, we've partnered with, I think we're the second company out of India to partner with GA4GH, which is a global organization which is bringing about standards on genomic applications, where we're kind of trying to see if globally where we can come up with standards on how microbiomes can be reported, as well as new consumer tools through which the data can be understood easily, whether it's traffic light signaling kind of reporting, or actually giving out values. I mean, I'm sorry, on the far right is a list of probiotics, and you can actually see that there is reds on both sides, on the left hand side as well as the right hand side. Too less of a probiotic is also bad, and too much of a probiotic is also bad. So we had to come up with unique ways of to showcase this. So it's turning this complexity into easy understanding and clarity is what we're trying to do, and which is how we've come up with things like a rich index, which is an easy to read score which anybody can read and say this is how good or bad my gut is. We also we realized that the functional aspect of the gut microbiome is very important. And not only with respect to probiotics and prebiotics, people are also now interested in what are the kind of short chain fatty acids that might be there. What are the kind of neurotransmitters that the gut might be producing at the moment. And combining all of this data is what we are giving out as a report. So taking confidence from all the anecdotal evidence that we got, this was our third clinical trial, and this was with type 2 diabetes patients. There were two cohorts, one with diabetic diet and the other one with a little more microbiome focused diet, and we got some very, very good results with it, with including immunity markers improving. So this gave us a clinical validation of what we're trying to do. And taking a cue from this, we've gone into multiple other studies. We're now doing a study on oncology with AIMS. We're also doing another study on FMT, which is starting pretty soon, and to try and look at different disease areas and see clinically if we can prove whatever we've been seeing anecdotally through consumers. Well, it'll make a big difference. Again, I don't know why some of my words are missing, but there are 4 problems that every microbiome product that needs to solve. The first one is actually is stability, and which is where the sample quality and sequencing and all of that kind of matters. The QC is very, very important over there. The second one is inclusivity, where we're looking at geographies and diets and of different people. I mean, there's India is so diverse in the kind of diets that they eat. I mean, it's and it all reflects in the microbiome. You give me a microbiome data, I'll be able to tell you whether this person is from an urban setting or from a rural setting, just by looking at the data. Whether this person is eating more rice or more chapati. That is the kind of information that you get out of it. You also get to see how much of capsules or tablets a person is eating, because even titanium dioxide is changing the gut microbiome. So there's very, very interesting things that is coming out of that. So having a large, diverse data set is very important. And lastly, in terms of getting accurate results, which is well established. So I think we're the first lab in India to get an NABL for the gut microbiome test. It was an interesting process. I mean, it took 4 days where the inspectors came. We sat and we had to educate them as to what we were doing. and to come to a consensus as to what the standards should be. And from there we've got a few ISO certificates as well, all to establish a good, credible test. And it all starts with the first step, sample collection. And in bioinformatics, they always say garbage in, garbage out. So if your data is not good, then your outcomes are not going to be good. Your data is only good if your sample that you get is good. And collection matters a lot. So these are the tubes that we're using recently. I forget the company's name. I mean, they're Bangalore-based. These are the tubes that we're using recently. So before this, we've tried a lot of the European tubes. We've got tubes from Canada as well. And, we tried a lot of them. There was one German company that we had settled down, and we were using it for about 3-4 years, and it worked well for us. And then I met Fatima, and the product spoke for itself. I just had to do one test, and I knew this was a good tube. And it's not the liquid inside. I don't want to belittle the liquid inside. I'm sure there's long hours of R&D and quality check and all of that goes in. But it's also the surrounding. The tube is stable. The screw never comes out. These tubes are thrown around in the remotest parts of India, and we're getting closer to lunch. I don't want to talk about how fecal samples are collected in India, where it is a brutal environment, and these tubes withstand that, the logistics and the QC. We have never failed a QC test with Azooka tubes, and that's very, very important for us. I'm still surprised that Alex and Fatima invited me today with the amount I torment them. Every day I'm giving them a new feature saying that when the cap rotates, please give me a signal that somebody's opening the cap, to things like, I want both DNA and RNA to be stable in the same liquid. But these tubes are excellent. And which is why, with confidence, we could get into the other microbiomes. Alex was talking about skin, and for the last couple of years now, we've been developing the consumer version of the skin microbiome test, and this is what we've launched, and we're getting some really good results. Especially the facial skin, we're seeing the microbiome actually plays a role in that, and we won't be able to do it without a good collection kit. And they were grateful enough. I'm grateful enough that Azooka actually developed a good one for us. Similarly, we're getting into the oral microbiome as well as the vaginal microbiome, and there's a lot of use case for that. Guys, the next one I need a scalp. There's a lot of requests for that. So different parts of the microbiome is what we're testing right now. Sorry, again, words are missing from my slides, but the other thing that I'm very passionate about is the Indian ecosystem that is building around this. We've developed the analytics, so that's a good part. Secondly, instead of importing tubes from Europe, we're now getting collection tubes in India. The 4 pillars that were supposed to be there in the box was collection, library prep, sequencing, and analytics. So the collection has been solved now. We're starting to see good library prep reagents being manufactured in India, which is a good sign. And I would say in about a year's time, it'll come up to global standards. DNA extraction is another place, another company down the road, Cambridge, which is doing really, really good work on doing DNA extraction. They are solving that kind of a problem, and of course analytics. So all of this combined is making the Indian ecosystem very robust. And the vision that I have and the ask that I have is, India should be a global leader in microbiome space. I mean, I'm so glad that the Kerala government has started the center of excellence for microbiome, and Bacillus subtilis has become the organism of the state. And this is a good initiative, and India should thrive to become the global king of the microbiome world. But it is also with this confidence, with the ecosystem that is now building, that we are able to ship our kits and our tools to the rest of the world. I'll tell you an interesting story from Brazil. So initially, our partners in Brazil were insisting that they were going to get tubes from Europe, and they tried it out for a couple of months, and then came back and said, maybe we should try your tube. I think it seems like it'll work better. And now instead of Blue Dart, I'm calling FedEx and saying I'm importing poop samples from Brazil. Please help me out. So yes, the tubes are going worldwide. Our microbiome technologies are going worldwide, and it is with great pride I can say that the ecosystem in India is building out great products. So it's not only that, we are also looking into microbiome-based therapeutics. Like I said, we've started a clinical trial on FMT. We're doing a lot of donor selections right now, and should get into the actual trial pretty soon. I'm from the drug discovery background, so anytime I get data, I'm always digging into data to see if there's anything that can be targeted in terms of drugs. So that is something that we're constantly doing. So with this, I'd like to end my talk. Again, a big thank you to Azooka for supporting us through our journey. From a small company, now we've become leaders in Asia, and this couldn't have been done with the excellent support, without the excellent support that they've given us. Thank you, and happy to answer any questions you may have. Yes.
The microbiome of an individual, will you get the similar results if you do the test on different days?
The answer is both yes and no. There is a small fraction which differs on a day-to-day basis, but the macros change over a period of 3 to 6 months' time. So we've done this. We've done a longitudinal study where we looked at people on a daily basis to see how much is changing. Yes, there are minor changes that happens, but from a health point of view, you got to look at. The change happens over a 3 to 6 months' time.
Hi, sir. It was a really wonderful talk. So I have a question in similar line. So when an individual is sick, that time his microbiome, overall the microbiome, whatever is there, it will be very different than a healthy person. Also, time to time, people will be changing their diet. So how do you basically come, like from one individual, whatever data you get, how much you can like value the data, because it might be very diverse depending on the diet and the overall health of the person from day to day basis.
Yeah, you're absolutely right. Which is why we had to collect a lot of diverse data sets before we could get into reporting. I think we spent the first 4 years, from 2014 to 2018, going around India collecting a lot of data before we could start the reporting piece of it. But the change is gradual. It's like I was just mentioning. It takes 3 to 6 months time to see a significant change in the microbiome, and although geography and diet plays a role, it takes time for that to actually seep into the gut.
Okay. Thank you.
Based on all the data you have an overall standard for healthy individual microbiome, which you can compare and diagnose something wrong with the person using the microbiome data?
Yeah, so I'll answer it slightly differently. We have data for what an unhealthy microbiome looks like. So I know what diabetes look like. I know what colorectal cancer looks like. Healthy is something, ideal and healthy is something that we have a basic understanding of it, but so that's
Is it different from the Western microbiome
It is different. It is different. The Western microbiome has very different phylums to the kind of microbiomes that we get over here. We also see a lot more fungal species in our microbiome, which is kind of rare in the Western microbiome. In terms of diversity, if you take an average gut microbiome of a young, healthy adult in India, you would see around 1,500 to about 2,000 species, different species. But as we age and if somebody has a chronic disease, that diversity comes down. What we still don't know is if the diversity increases to 2,500 to 3,000, whether that is good or not, that is something that we don't.
Yes. How about also using tear as you said urine and vaginal sample microbiome. What about tear samples? Because for many diseases, the tear is a very good marker in diagnosis and other various disorders. So I was wondering whether you are considering use of tear sample for analyzing the microbiome.
We're very early stages in that. I think we've done about half a dozen samples till now, tear samples.
Some small metabolomics we had done using the tear sample, and we were kind of surprised to see butyric acid there.
Correct.
Because it should come from the gut. So I was rather curious to find out if you had done any work in the tear samples.
We've just started it, so it's about half a dozen samples that we've done.
Okay, thank you.
Hi, my name is Madhavan. I'm founder of Theomix International. I've been following you for some time, so as a bioinformatician as well. So I guess two things you missed. Maybe you're already doing about it. One is about the nasopharyngeal component and upper respiratory. So these two, I personally feel there's a huge gold mine out there, because specifically talking about immunity and the defense part, right? And the general human habit about spitting and we do all this stuff, right? So sneezing and all this stuff, right? So the microbiome of nasopharyngeal and the upper respiratory holds a higher promise in terms of how much we could manipulate it so we get better immune existence, right? This is one of my weird thinking. So I'm not sure if you have accounted it right, as a translational part. So I'll be happy to discuss about it.
Sure. We published a paper where we showed that a better diversity in the gut actually helps fight off lung infections. So there's definitely a crosstalk over there. We'll discuss more.
Sure.
Personalized for one individual to another, and diet changes, how it will affect.
Yes, that's the study that we did. That the personalized microbiome diet is actually playing a role in making you healthier.
Kumar, I have a question. Now that you're going global, congratulations on all the work thus far. From the consumer experience standpoint, how is personalization going to happen when you go globally? Because obviously microbiome composition is going to differ. How has the consumer experience or the probiotics that you've given, is it standard across? Because it seems like a combinatorial nightmare.
It's not only the probiotics. So one of the reasons probiotics that are imported from outside India does not work in India is because the strains are very different. It just won't colonize the Indian gut. So one, at the strain level itself, it's very different. Secondly, at a diet also, there's so much of difference, right? So when we went to the Middle East, suddenly there were questions about camel milk. And, so we had to do a little bit of homework on things like that. So we are, as a team, very confident on diets that are there in the Middle East, Mediterranean, Europe, Western, because there's a lot of data. But if we have to go enter China, we won't be able to report because I have no idea what what snake would do to your gut, kind of thing.
I mean, just a quick follow-up question. So in that context, do you leverage existing data sets that are there publicly available, or what do you think would be a minimum data set that you can kind of need to enter a market successfully? I mean,
I would say at least about hundreds would be nice, because there's a lot of public data that is now available, and the R&D team right now is consuming 10 terabytes of hard disk space every quarter now. I mean, so that's the amount of public data that is available out there.
Hi, do you hear me? Hi, so this is Uttara Chakrabarti from Manipal Institute of Regenerative Medicine. I am extremely, kind of intrigued with all the data and so much of work that you've been doing in handling the microbiome. Coming specifically to talk about the oral microbiome, which is so diverse. So when it comes to identifying targeted therapy against specific species, for example, as you mentioned, more fungal species in the oral microbiome or in the Indian population, if I'm talking about, especially because I am interested in looking at providing solutions for the fibrosis, which is related to the spread of these pathogens there. What would your approach be in order to identify or rather single out the fungal strains from the bacterial strains in oral samples, and especially considering so much of diversity that remains in these different types of pathogens?
With the oral samples, the challenge right now, I think a lot more study needs to be done on that, because we see a difference between saliva and subgingival plaque. And I think getting into the subgingival plaque is a much better way to determine what's happening in the oral cavity than relying on saliva alone. And I don't have an answer to the therapeutic angle right now on this, but the oral microbiome is not as well studied as the gut is, and I think a lot more energy and time needs to be spent on this.
All right. Okay. Thank you.
Hi, sir. One more question. So, have you ever considered taking samples from Northeast India, because they follow a very diverse diet? Since I'm from Northeast, so I know that they follow a very diverse diet. So, probably if you can't go to China, but you can still go to Arunachal and collect samples, because it will be definitely very different data than rest of the Indian.
Yes, we have. I mean, we've collected samples from all the states up there.
Okay.
It's probably not as rich as the rest of India in terms of the number of samples, but we have collected samples.
Okay. So, do you see any difference in terms of your data, like that?
There are differences. I mean, the one thing that you can very clearly make out, especially the easternmost states in the northeast region, is that there's not too much of industrial pollution that's actually entering the gut data, which is not surprising. It's not as urbanized as some of the cities of here.
Okay, thank you.
Thank you so much, sir. I must say you truly delivered both in substance and in style. I would like to call upon Kumar Murthy, sir, to felicitate Kumar, sir. We will now have a small tea break for about 15 minutes. Please join us for refreshments outside. The washrooms are on to your left side outside the door. Thank you. His impact goes far beyond the clinic. A lifestyle medicine practitioner and global speaker, he blends medical science and real world living. From serving as a wellness consultant on international cruise lines to leading John Maria's wellness group, his work sits at the intersection of prevention, healing, and empowerment. Fluent in multiple languages and delivering over 100 interactive sessions every year, Dr. Manoj is known for turning health conversations into engaging, practical experiences. Please join me in welcoming him.
Hello. Hi. Good morning to all. I'm Dr. Manoj Johnson. I'm a functional medicine practitioner and I'm a naturopathy physician. And we have 17 clinics in Kerala. Especially we are focusing on gut health. I'm very happy that in previous session, most of the people are very much interested in gut microbiome. So this is a therapeutic part of gut microbiome. So we have more than 60 plus doctors from the different medical system. We have a good protocol for treating autoimmune and chronic conditions. And we are treating with the probiotics, prebiotics, and evidence-based therapeutic herbal medicines. We are treating per day 500 to 800 patients. And we have more than 4 lakh database since we are practicing since 10 to 12 years. And I'm not a scientist, I'm just a clinician. So today I'm going to present my experience. The role of gut microbiome in autoimmune and chronic diseases. So already the previous presentation, the Shankaran sir already explained about gut microbiomes. The technical part we already covered. So this is the therapeutic part. What is gut? As we know already, gut is mouth to anus. Not only the intestine, from mouth to anus is gut. So we have 30 trillion human cells in our body, but we have 100 trillion bacterial cells. So basically, when we talk about gut microbiome, disease conditions and health conditions, usually we tell about good bacteria and bad bacteria. It is not about good and bad bacteria. It is the shifting the location from one place to another. So we have a H. pylori in everyone's body, but if it is increasing the number, then it is bad. Methanogenic bacteria, whatever the bacteria, if it is in the normal range, it will be good. But if it is exceeding and if it is shifting from one place to another, out of 100 trillion gut bacteria, it is a 99 trillion gut bacteria located in the large intestine. Only the 1% in the small intestine. So there are two types of bacteria, aerobic and anaerobic bacteria. In the colon, large intestine, most of the bacteria are anaerobic bacteria. But in the ileum, jejunum, duodenum and all, anaerobic, aerobic and anaerobic and aerobic. So in the stomach is anaerobic. So these are the major two types of bacteria in our gut. So bacteria are our bacteria are not the not enemies. They are our friends. Basically, when we do the dosa, the bacteria should be there to convert. If it is alcohol, the bacteria presence should be there. Pickles, the bacteria presence should be there. From milk to curd, bacteria should be there. Then only it will convert. But usually, without bacteria, our food will decay. It will not ferment. The same way, The environmental, in the like bacteria will ferment or decay the environmental waste. The same way in our human body, it will ferment, recycle, absorb, and eliminate. This is the major role in our gut. So we have, I already mentioned, we have 100 trillion bacteria, but only few bacteria, each bacteria has its own function. So the major thing, lactobacillus produces lactic acid, maintains acid gut pH. Bifidobacterium enhances the gut lining protection. Akkermansia municipalia improves insulin sensitivity. So most of the insulin resistance condition, it starts from the gut. So we usually tell fatty liver is because of insulin resistance, diabetes is because of insulin resistance, PCOD is because of insulin resistance, obesity because of insulin resistance. 80% of the insulin resistance starts from the gut. And the raspberry and prochella, that is, it produces small chain fatty acids, that is butyric acid, in our gut, that is anti-inflammatory. So the gut-brain connection, 2020, in Nature, published in Schizophrenia Journal, the role of the gut microbiome in the development of schizophrenia. That means they transplanted the schizophrenia fecus of gut microbiome of schizophrenia patient into a mice, and within few weeks of time, the mice developed same schizophrenia like symptoms. So most of the people, they are having behavioral disorders, autistic related problems, hyperactivity problems, irritability, uncontrolled anger, emotional disturbances, depressions, anxiety. It is 80% connected to gut. So gut related diseases, one is autoimmune disease. That is 60% of the autoimmune disease starts from the gut. So that's why most of the people are taking treatments for autoimmune. They can't stop the medicines because it is not addressing the root cause. So another one is digestive disorders, as we know already, bloating, gas, gastritis, IBS, diarrhea. Most of the people are having bloating problems, gas productions. That is, their gut doesn't have the capacity to digest the carbs and the fibers. So most of the people don't know whether they can consume the carbohydrate or not. Because for gut related problems, fat is comparatively better, non-veg items comparatively better, but not the carbs and fibers. But the carbs and fibers are good for health, but we should know that in our gut, we have the capacity to digest or ferment that particular bacteria should be there. Then only we can consume those type of food. Brain disorders, that is psychiatric disorders, behavioral disorders, emotional and mood disorders, neurodegenerative disorders, especially Alzheimer's conditions and Parkinson's conditions. And 60% of the Alzheimer's and Parkinson's problems are connected to the gut. Allergic disease, most of the people are having sneezing, especially in the morning time, eye itching, ear itching, dandruff related problems, dry skin, eczema conditions. This is 80% of the allergic conditions are connected to gut. So how the oral bacteria is connected to autoimmune disease. 60% of the rheumatoid arthritis is gut mediated. Gut mediated means there are two major bacteria, one from the mouth, another from the intestine. From the mouth is gingivalis. That gingivalis bacteria, some people having gum bleeding or gum infections. That bacteria will produce the chemical. This chemical that will convert L-arginine from the joints to L-citrulline, that is the foreign body. Because this bacterial producing foreign body in our joints, and the body will consider that chemical like a foreign body, and the immunity will damage the joints. But usually for rheumatoid arthritis, the doctors will treat only the joints, but 60% of the conditions, that is around 30% is oral, and next of the 30% is gut, that is Provotella copri, that is another bacteria. So Ayurveda, in the ancient times, Ayurveda used to tell Amavata. Amavata means it is completely related to gut, but it's not completely, but 60% of the rheumatoid arthritis is connected to this particular bacteria present in the gut. This is the study published in Frontier, relationship between Prophyromonas gingivalis and rheumatoid arthritis, a meta-analysis. Psoriasis, this is my favorite topic because in our clinic, most of the people are coming with the psoriasis condition. In that, see, 70% of the psoriasis is connected to gut, not 10, 20, 30%. It is 70% of the psoriasis condition. And the rest of the 30% is related to oxidative stress, something related to liver, thyroid, and toxicity, so many other reasons. But that is 30% is non-gut mediated. So how we can confirm whether it is gut mediated or non-gut mediated? Because whoever comes with the psoriasis condition, first we used to do the test. It is called one IG, immunoglobulin E, that is a blood test. If it is connected to gut, the infection is connected to the gut, means 100% their IG level will be high. If it is a normal range is 100, the psoriasis condition, if it is connected to gut, their IG level will be minimum 1000, 2000, 3000, above 500, not 200 or 300. It will be above 500 to 5000, 10,000. So we can easily make out it is connected to gut. And another one, no need to test, no need to do any blood test. Without doing the test, we can confirm whether it is connected to gut or not. It is very simple. That is it's an Ayurvedic method. It is a purgation. Taking enema, and after cleaning everything, the next day the flakes is reducing, then it is connected to gut. If it is not related to gut, then the flakes will not reduce. So within 15 days to 3 months, we can reverse the psoriasis condition if it is related to gut. So that's why psoriasis is very easily we can treat if we know the root cause. Because most of the doctors, the medical systems are, and the common people know only psoriasis is skin disease. It is not at all a skin disease. It is autoimmune. It is something related to internal organs. And the migraine, this is my another favorite topic. Most of the people are having severe migraine headache. The migraine, this is my experience I'm telling. Migraine is mainly because of 3 major reasons. I'm not explaining the explanation in medical textbook. Basically 3 major cause for the migraine. The first cause, root cause is H. pylori bacteria or gut dysbiosis, that is related to stomach. Second reason is hormonal imbalance, especially in ladies, the irregular periods or PCOD problems or bleeding related problems, weight gain, so many hormonal imbalance problems. If they don't have any stomach issues, still they have hormonal problems, then we have to treat the hormonal imbalance, then automatically the migraine will reduce. And if the person don't have a stomach problem and don't have a hormonal imbalance, then it is 100% connected to neuro-related problems, the poor circulation to the nerves. So the basic mechanism is 60% of migraine is connected to gut. So how the migraine mechanism? It's very simple. we have a vagus nerve. It is connected from gut to brain. If anything irritating in our gut, especially the H. pylori bacteria or any other infections, it will irritate the nerve endings, and this irritation will pass through to the brain. Here in the trigeminal ganglion, we have a, this is the mechanism. I'm not going to explain the mechanisms, but the ultimate reason for migraine is dilatation of blood vessels in the brain. But the one stimulation is from the gut. If the person is not taking food in the proper time, it will aggravate. And when the stomach upset, it will aggravate. So if it is connected to the gut 100%, that nerve will irritate, blood vessel will dilate, they will get the migraine. So it's very easy. If it is connected to gut, we can treat very easily migraine, because the irritation, if the irritation is reducing, then automatically migraine will reverse. Not reducing, I'm telling. In the future, they won't get the migraine attack again if it is connected to gut, because we are focusing more into the gut aspect. So as we know already, if it is jar, if we put cut vegetables inside the jar and put some water, within 3-4 days of time, it will decay. Is it? We know already. But in the same jar, put the cut vegetables and the water and add little vinegar, but it won't ferment, because the bacteria will not grow in the acidic pH. The same way, why people are getting more skin related problems, autoimmune problems and chronic diseases, it is shifting the bacteria from one location to another location. It should be there in the large intestine. It is shifting from large intestine to small intestine. That means someone from the security office, he should be there in the security area, that front of the building, he should be there in that area. But if he is sitting in the MD, managing director's chair, that means he is good in that place, but he is not good in managing director chair. So the same way, the bacteria is, if it is located in the large intestine, then it is okay, it is functioning normally. If it is shifted to the small intestine, then all the problems will start. So the same way, so what we have to do, we have to reduce the growth of the bad bacteria. Then only, if someone having eczema condition, or as we know already, the varicose ulcers, the varicose veins and varicose ulcers are totally different. So varicose veins are, so many people, 60% of the people, they have a bulged varicose vein. We can see clearly the vein bulges. So 40% of the conditions, internal varicose. When we can understand whether it is varicose means, at last the discolorations will be there, swelling will be there, pain will be there, and sometimes itching, and the blood will come out. So next, it leads to ulcers. So, but 60% of the conditions, varicose veins are okay, normal. It is not connected to gut. But sometimes most of the people, when the skin color is changing, then it is connected to gut. In that 60% of the people, they can't digest the casein from milk. So they should not take milk at all, those who are having varicose ulcer condition, varicose eczema conditions, because it is not digesting properly. Not only milk, some of the fishes and shellfishes and peanuts, and 30% of the people having gluten intolerance. That is another type of chemical component. So this is because of the gut problems. So whoever treats the varicose ulcer without treating the gut, they won't get the result. The same way, the autism and hyperactivity condition, the parents will focus only the behavioral therapy and play therapy. So many therapies they are focusing, but the 60% of the conditions, gut is we have to treat the gut because the Clostridium group of bacteria is the overgrowth that leads to autism and hyperactivity problems. So what we have to do, we have to clear the bad bacteria, overgrowth of bad bacteria in our gut. That is only one solution, that is small chain fatty acids. So we can't put vinegar into our intestine, but still how we can get the result? It's very simple. Prebiotics, not the probiotic, prebiotic. Most of the autoimmune conditions, autism, hyperactivity, skin conditions, joint inflammations, emotional disturbances, when we treat the large intestine and the small intestine especially, we have to focus, we have to reduce the growth of bad bacteria in our gut by adding prebiotics in our diet and therapy. So these are the prebiotics, because we are eating only for 30 trillion bacterial cells, that is our human cells. But we have 100 trillions bacterial cells. For the bacterial cells, we should eat for bacterial cells. That is, the prebiotic is the food for gut bacteria. So these are the major food items of prebiotic. But in our clinical practice, therapeutic purpose, we don't give this because this is not at all enough. This is for the maintenance. This is not for the therapeutic purpose. Therefore, the therapeutic purpose, we have a prebiotic fibers, different types of prebiotic fibers, inulin, gum arabic, different types of prebiotics. When we add in the treatment, we are getting excellent result for autism conditions, especially hyperactivity, irritability. Children's there, no, they will walk, shout, they will destroy so many things. We are getting excellent result for within 2 weeks, 3 weeks itself. So along with this, they can do all the therapies, no problem. Only with the therapies, it's very difficult to get the speed recovery. So with the treatment, we are getting the excellent result. So the probiotics, for each and every condition, different probiotic. If you have a hard stool, then it is different probiotic. If it is loose stool, different. Initially hard stool, then loose stool, different probiotic. If it is hyperactive and autism condition, different probiotic. Constipation or hard stool is not because of less fiber or dehydration. It is because of overgrowth of methanogenic bacteria. The bad bacteria is problem. So if it is fiber or water is the main culprit, then imagine lion, tiger, cheetah, they're not taking any fiber items, no fruits, nothing. Still they don't have any constipation. But we should know that in our gut microbiome, whether we can eat this particular food or not. This is proper consultation should be there. Without knowing what I can eat, then all the problems will start. Most of the people are having allergy problems. First consultation itself, we used to tell, avoid gluten and lactose. And if still they have skin allergies or itching or problems, then we will tell they should avoid tuna. That milk is 100% we should avoid. There is no other option. Especially they can take almond milk or coconut milk. The plant-based milk is comparatively better, but the animal-based milk, 70% of the conditions we tell you should not take milk at all. If you have bloating, constipation, hyperacidity, those type of problems. Because when you take the pantoprazole or ranitidine, whatever it may be, you have to take for lifelong. If you are not taking one day, it will aggravate. Because when we take the milk, you won't feel much changes. We focus only spicy items, sour items, pulses, because that is a highly irritant. That's why we focus only that type of food items. But milk has so many problems because most of the people, they can't digest milk at all. So those are having skin related problems, thyroid related problems. How this gut and thyroid is connected? Hypothyroidism is It won't come suddenly. First thyroiditis will come, and afterwards 5 to 8 years of inflammations. Then at last the hormone production will reduce. That time we will supply the thyroxine, but it will balance only the hormone production. Rest of the problems and inflammations, what and all functions the thyroid is supposed to do, that everything is impaired. Because just to see, those who are taking hypothyroidism medicines, especially thyronome or thyroxine, their TSH level will be always normal, but still they will get hair fall, still they will gain the weight, still they have the inflammations, pain, tiredness, sleep disturbances, anger problems, miscarriages, so many problems. These are all thyroid related problems, but when they check for the thyroid, it will be normal. So correction of hormones is not the proper solution for thyroid conditions. It is an inflammation. That is 70% is connected to gut. So basically, I'm not a gut person, but I used to treat since so many years, 10, 12 years. But at last, how I came to this particular area means it's very simple. When I treat the gut, they'll tell after 2 weeks, they'll tell my sleep disturbance is gone. So because I didn't treat for the sleep problems, they'll tell I had neck pain and back pain. Now it is reduced. I had severe migraine, reduced. I have severe tiredness and fatigue. Now it is okay. So then I started, why it's coming like that? Because if someone is coming for the sleep disturbances, usually the doctors will give sleeping pills. But when you treat the gut, automatically it will reduce. Because if the iron absorption should be there, then only they will get the proper sleep. Vitamin D absorption should be there, then only they will get the proper sleep. If gut is not functioning properly, the estrogen will not excrete through the feces, it will reabsorb. Again, the fibroids, ovarian cyst, it will form. Just imagine those who are having the fibroids, when you do the surgery for the fibroids, again it will come. After few years, again it will come. Because the root cause is still the estrogen is reabsorbing. It's becoming more and more in our body. This estrogen leads to fibroids. So it's very simple. When we treat the root cause, we are getting the excellent result for so many problems. The irritability will reduce, the sleep will okay, and the proper absorptions will be there. Most of the people are eating normal foods, healthy foods, still they have so many problems. When we check the vitamin D, it will be very low, but they are taking vitamin D supplements continuously. When we check the B12, sometimes it will be very high, sometimes it's very low. But this is the problem of gut absorption, especially the iron. Most of the ladies, they have iron deficiency. It is mainly because of the gut related problems. So we have to clear the gut dysbiosis, then automatically. So we have a gut reset therapy for so many problems. And first, they will tell 8, 9, 10 problems, different symptoms they will tell. In that, we ask whether you have constipation or bloating or acid reflux, anything. If they mention anything related to gut, we focus only gut. And we'll tell, okay, after 15 days, out of 10, how many problems reduced, how many remaining? Just tell me that much. Because if out of 10, minimum 5 or 6 will be reduced within 2 weeks of time. Then rest of the things we have to treat mainly for their problems. So this is my experience. That's why I started. Now I'm a PhD scholar. This almost third year, and I focus more into my PhD subject is gut microbe and thyroid connection. How these small chain fatty acids will affect the thyroid functions, thyroid gland. So, but this is my subject. But I used to deal with all the autoimmune and chronic conditions. So this is the grain free, because most of the people having gut related issues, we supply the probiotics, prebiotics, digestive enzymes, herbal medicines. This is the combination, not only one Ayurveda or Homeo or Unani or modern medicine or something, not like that. We combine all the medical system together. Most of the people are having fungal overgrowth. If the fungal overgrowth means they will get dandruff related problems. They will get bad breath, smell, sweat smell, and iron deficiency. That is related to fungus and the parasite overgrowth. So we should treat this one, then only we will get the result for bad breath and dandruff. So this is the outline. So the summary is, when we have chronic disease or autoimmune or any other skin conditions, emotional disturbances, tiredness, whatever, we should focus, we should we should fix the gut, then we'll get the excellent result. Thank you very much for inviting me here, Azooka team. Thank you.
The floor is now open for Q&A.
Yes, prebiotics.
What are they called?
We have different names: Gutwin, Prebiotic Plus, Gut Care. This everything related to prebiotics. Different combinations.
But they're sold in the.
Yeah, we have a e-commerce website, and we have 18 clinics, and one clinic in Indranagar in Bangalore. Basically, I'm from Karnataka, but my all establishment in Kerala. In Kerala, in every district we have clinics, and we treat with the prebiotics, probiotics. We have our own brand, different bacteria and prebiotic fibers.
Have you done analysis on how this fares compared to other brands of prebiotics or probiotics available?
Yeah, if it is Bulradi, that is one special category of probiotic, that is spore based organism. Most of the probiotics, when we eat, when we swallow or when we consume, it will because of the HCl acid, it will destroy most of the time. But it is a spore based organism, means it will reach to the small intestine or large intestine. It will, because the acid will not destroy the condition. So we have a different form of probiotics. Some it should enter only the stomach area. Some it should reach up to intestine, up to large intestine. And most of the large intestine conditions, no need to treat with the probiotics, only with the prebiotic. So that's why more than probiotic, prebiotic is the ultimate solution for most of the autoimmune and chronic condition.
Hi.
Yes.
Yeah, this is Mati. Since you, that's a good presentation. You mentioned about the anemic. Sorry. So you mentioned about the anemic condition, right?
Yeah.
So we did some small study on the Indian population, where mostly the women, pregnant women especially, and the female kids, right? So they are mostly anemic. So we were into the nutritional aspect, how the food is absorbed or it's not unavailable in this kind of a female population. So since you mentioned that gut microflora, that is also the major reason, right? So,
yeah.
So how do we treat that way? Because the food which is consumed in the home usage is the same, right? For a male or a female, right? So in that way, as a common food culture, what we have in India, in 5 people, 2 female, the mom and the kid, and the dad and then another male kid. So in that way, what is your suggestion? How to improvise that?
Okay, first thing is, we are not same. So when we see the diesel car, petrol car and the electric car, it looks same. 4 wheels, steering, metal body, the purpose, everything is same. But the source is totally different. So if it is husband and wife, means they are genetically from the 2 different family. So whatever we are eating, one person, if the same gene means it's okay. If it is another different gene, means it is totally different. The source is different. If it is gut microbiome, if it is parasite overgrowth, means whatever food you take related to iron content, the parasite will eat most of the time. And the second one is, when we see the iron rich food, only few iron rich foods are we have bioavailability and absorption. So we tell so many food products, but there is no use at all. You know, moringa leaf. That moringa leaf is very good source of iron. But when we check in the Google, what and all the high source of iron means, they have very big list. There is no use at all. And the non-veg item, especially the mutton or beef or liver organ meat is high source of iron content. So only these three I found it's a good source of iron. That so many plenty of food items are there. There is no absorption at all. So we are thinking that we are taking all healthy foods, and this doesn't have any absorption, because most of the people are having this gut dysbiosis and gut related problems. Some people are having a bacteria or fungus overgrowth, bad bacteria overgrowth. So we have to analyze properly, and then we have to tell the diet. Then only they will get the result. Because iron is very, very important for, especially for ladies, for the hair growth, for the menstruation, for the thyroid condition, for skin, for sleep, for stress. So whoever comes in my clinic, I'll give the herbal combination of iron, because I don't give the chemical combination, because so many people are having gut related issues after they take iron tablets, and constipation, or so many problems will be there. So the herbal, that's why I told you, herbal medicine is comparatively better than normal iron supplement. When we supply the higher dose of iron, within 2 weeks, weeks of time, they will get the sleep. They will get hair fall will reduce, tiredness will reduce, palpitation will reduce. So that is totally different. We can't tell for one huge population this is good, this is bad. This is based on their gene, their gut health, their stress level, their body nature, their lifestyle. Then only we can tell this is good or this is bad for you. Thank you.
Hi, great presentation. So I would like to ask that in your treatment practice, do you use any kind of allopathic medicines, or is it completely herbal?
Yeah, no. Allopathic medicines are there, but not in all conditions, because most of the people they already taking so many medicines, and they fed up of that, and they are telling that I am taking this medicine for so many years. I want to get rid of this. So in that time, we don't give the PPA, like ranitidine, pantoprazole. We don't give that, but we focus only. Sometimes we give the vitamin B12 injections, and sometimes albendazole, ivermectin for deworming conditions. Because when we give something, it should absorb. Before that, we should clear the gut. For that, we include all the type of medicines together. First, this enema also good for so many things, not for all. Only 40% of the conditions we will tell you should clear from your gut. Then deworming. That is, some people having albendazole and ivermectin combination is more than enough, depending on their body and weight. But some of the conditions we should give 3 days, 5 days, 7 days for different protocols. So we give all the, we have, that's why I told you, we have 60 plus doctors from the different medical system. We have a strong protocol. We can't treat only with the one medical system. Or one medical system, we have limitations, we have positive things. So we combine all the positive things together and we treat, we get the excellent result.
Oh, that's great. And you follow up with blood tests and
Yes, full blood test. And 60% of the conditions we can find out with the blood test. But we do 100% we'll do scanning also. My gut microbiome test, saliva test. Those who are economically okay, they can bear the fee. Then we can go for stool mapping, something like that. But most of the conditions, scanning and blood test is more than enough. But not the normal test. When we do the normal, when we check HB, hemoglobin, no use at all. We should check iron and ferritin. Always the HB level will be normal, but they'll get all the anemic symptoms. So the iron and ferritin is very, very important. If it is B12 is from 200 to 900 is normal, it should be there above 600. Then only they will get the result. If it is vitamin D, if it is maintaining around 30, if 30 to 100 normal range, if it is maintaining 30 means they will get result for bones and muscles. If it is maintaining 60 level, they will get result for allergic conditions. If it is level maintaining at 80 or 100, they will get excellent result for autoimmune conditions. So it's not about normal. Depending upon their health condition, we should maintain in that level. Then only we will get the result. Or else, when we check the blood report, everything will be normal. But still, they have all the problems.
Thank you so much. Hi. I just want to ask that what is the level of impact of stress and mental trauma on someone who is otherwise healthy on the gut microbiota?
Yeah, that's why I already mentioned. Most of the people, they focus exercise, proper diet, sleep. They try to sleep. They focus more healthy lifestyle. Still, they will get most of the problems because if the gut microbiome is not functioning proper, gut dysbiosis is there. The gut bacteria will produce the serotonin. The basic function of serotonin in the gut is movement, that peristaltic movement. And after that, this same serotonin, it should enter to the brain through the vagus nerve. If it is not entering, they will get irritated and they will get upset for small things and they won't get the sleep. The same way, if the stress level is high, again the gut will irritate. So these are connected. Most of the people, when those who are having gut issues and thyroid related problems, 100% they will get stress, anxiety, depression, irritability problems. So we have to focus the root cause, then only we can reduce the cortisol or stress level. Or else we should take anxiety medicines for anxiety or depression. So meditation is not at all enough for those condition, because that is a chemical imbalance. We should clear in where the root, where the problem, whether it is a gut or thyroid, we have to find out, then we have to treat.
And how long does it take to restore the imbalance of the gut? If
minimum 3 months. But within 2 weeks itself, we will feel the changes. Every 2 weeks we used to follow up. Then after that, if it is not getting proper result, then we'll change the protocol.
Hi,
Yes.
my name is Madhavan.
Hello, sir.
You represent clinical part, so it's nice to ask a question. How much time typically it will take to have the gut microbiome reset after prolonged exposure to antibiotic?
See, when we take the antibiotic, so the damage will be there from minimum 1 week to 3 months. And within this type of this time, we have to clear with the probiotics and prebiotics. Then only we won't get in the future problems. So most of the time after the antibiotics, we don't focus much about the repairing. We just kill all the bacteria in our gut, and we'll get the relief. Yeah, but we should put the good bacteria in the particular area. So that is minimum 1 week to 3 months. We should reset the.
But really, there should be a natural way of reconstruction, right? Everyone's body should be able to rebuild the microbiome by itself without any supplement of a probiotic, right? Because assuming that, we talk about probiotics today.
Yeah, there is a natural process.
It's a natural process, right?
But those who are having the health issues after that, like skin related problems, severe joint pains or muscle inflammations, tiredness, irritability, sleep, those type of problems, no? That means they have the problem.
So the indirect question to that is that do you recommend giving probiotic recipe for everyone who takes a course of antibiotic?
It's not necessary, because some people have a food poison, like Shigella bacteria or Salmonella. This bacteria will enter and it will show so many problems in our gut. But usually we'll take antibiotics or something and it will reduce. But because of this bacteria, this bad bacteria, body will produce antibodies against this particular bacteria. That bacteria will die, but the antibody still it will be there. This antibody will irritate the gut again and again. When you check for the H. pylori, when you check for the salmonella, most of the time won't be there. But still you feel the same problem. So we have to clear from the root, that we have to clear the antibody itself. Then only we'll get the result. So that's why it is not necessary that those who are taking antibiotics, they should focus probiotics. It's not necessary. But those who are having repeated gut infections, food poisoning, and like skin related problems, they should reset the gut after antibiotic course.
Thank you.
Okay. Thank you. Thank you very much.
Thank you, Doctor. That was deeply insightful and relevant to our holistic health benefits. I would like to call upon Dr. C.V. Natraj to felicitate Dr. Manoj. For our next speaker, we have the world-renowned biochemist and one of India's leading women virologists. Dr. H.S. Savitri began her journey at the Indian Institute of Science as a PhD student and went on to become the first woman faculty and the first woman chair of the Department of Biochemistry. Over decades, her work in plant virology and molecular biology has shaped how we understand virus architecture and genome expression. She is also the first woman to receive the Karnataka Rajyotsava Award, a testament not just to scientific excellence, but to breaking barriers with quiet strength. Today, she continues to lead translational research at Vitala Eye Hospital, proving that science for her it has always been about impact. Ma'am, it's truly an honor. So please join me in welcoming Dr. Savitri.
Thank you. Once again, I'm very happy that I'm here. Thank Fatima and Alex for inviting me for this exciting talks. I have really thoroughly enjoyed every talk that was presented. Now, Alex and Ben are very, you know, they just wanted me to speak, although I have not done much research for the past 10 years because I retired 10 years ago. But I just thought I'll share with you the beginning of Azooka and how I was involved in the beginning of this journey that has happened since 10 years. Next slide, please. Now, the title was Safety in Biological Research. When you talk of safety, there are many aspects of safety in terms of how you do research and how safe what you are doing is. And for most molecular biologists, it's important that we minimize use of chemicals that are carcinogenic, that cause mutations in our DNA, or they are cytotoxic. And world over, all molecular biology labs use the cybergreen or ethidium bromide for detection and quantitation of nucleic acids. Next slide, please. So now there is a gap that here we have to find some solution in terms of being able to find a molecule that is not carcinogenic and that can also replace these world leaders. So the most widely sold DNA dye is the cyber green, which is used in all RT-PCR and other DNA detection techniques. And most of them are intercalators, that is, go in between the bases of the DNA and distort the DNA, and finally it can lead to mutation. Next slide, please. Now, I think I had asked for change in the order. Somehow the same order has come. Anyway, there are some scientific limitations to the existing dyes. That's what Fatima had gone through the literature and found out that most of the dyes that are currently used in the market are DNA intercalators, and then they are uh, you know uh, compatible, but many of them with PCR methods, but some of them are inhibitory in nature and they're hazardous. One has to find a very good way of disposal. Just putting them down the drain would make the water also have these carcinogenic substances. So there is a lot of safety issue involved in using these dyes. Next slide, please. So I just got into the problem of these dyes through Fatima. Fatima had found a dye that was from a plant source that could stain nucleic acid. In fact, I really like her, passion and perseverance for doing something that she started with for her PhD, because often we do our PhD in some subject and then move on to do postdoctoral in some other subject and things like this. But with one-minded approach, she wanted to continue the discovery that she has made. At that time, during her PhD, what she had shown was that a plant extract can be used for staining DNA. Then she had to show or establish whether it can become a product or not. So that is when she came to my lab. So next slide, please. So you see, what it entails is to start with the extract and start purifying the compound, because no, you cannot market something which is inconsistent in its source. For example, batch to batch variation. You get a lot of batch to batch variation if you were to use this the extract itself for staining purposes. And for scientific credibility and for acceptance in the scientific community, it is important that the compound responsible for the staining purpose is identified properly. So when she came to my lab, at that time she had this idea of what to do and all that. And so Alex was there as her co-investigator in terms of trying to find how funding for identification of the compound, and then moving on to making synthetic dye equivalent to this cyber green that can be sold. So they wrote their first big BIG grant, and as usual it was rejected, not because it was scientifically not satisfactory, for various other reasons. Then they went to STEM, and then they were about to see how they can continue with the work, and it so happened that for some reason that the person who was there in the big grant was saying that, why don't you sell your idea and take some money and be happy with it? Why are you struggling? Why do you want to go through this entire identification and all that? It is a long road, you will not succeed. But I must say that to the credit of Fatima and Alex, they said, no, we are not going with InSTEM because we don't want to sell we want to innovate, we want to do it something here itself, not give it to some foreign company and be happy that they gave us some small money. So then I said, why don't you go and approach Nataraj? He's really good in this. He's going to help you with this. And there they began their interaction with Nataraj and trying to see how they can get a small grant from SID and start working on the identification of the molecule responsible for staining the nucleic acid. So uh, at that time, luck also, beginner's luck or whatever you call, is needed. There was a MSc project student who had come to my lab. I asked Fatima, do you want to take him and start some, identification and characterization of the mode of binding of the DNA, etc., etc. And then it turned out that Abhishek, who was the MSc student, was really dedicated, and he was able to show that the extract, the DNA, the compound can bind to the DNA through the minor groove, and it is non-mutagenic as it's from a food grade plant product, and therefore all these things. And then quantitation of the bound DNA through fluorescence, it had a very high quantum yield. This took 4 years for them to establish what is the mechanism by which it binds. In the meantime, Fatima was, through Nataraj, was able to find a very good chemist who could, meanwhile, they had extracted the compound and analyzed it by spectroscopy, and they got the structure, and there was a very good chemist who made the synthetic product and made several derivatives. So you see, research doesn't happen in one area. You need a lot of expertise in different areas, and thanks to this very good chemist who was able to make so many derivatives, and there it was a lot of work. So some two more people, Vandana and Anjana, came in, and then Harshini, who was in my lab, came in, and they all chipped the work and tried to establish the mechanism by which the dye binds to the DNA, and also how it can give a very high quantum yield. Now, at this time, we were struggling to see how we get consistent results and how we can, emphatically say, yes, this is a very good product and it can compete with ethylene bromide and cyber green, or it's better than that. At that time, COVID struck, and they also had a great difficulty in raising money, investors, and finding money for further work in this Tintorang, because it was very promising, but at the same time there was no funds and they were running out of money. Sometimes, something happens like blessing in disguise. COVID-19, I had to shut down my lab, and therefore Fatima had to, come under great, observation and supervision and carefully take away all her samples from my lab so that she can start something on her own. Again, SID and Nataraj were instrumental in seeing that they can relocate to another. See, all this time they were working in my lab. Suddenly the lab closed and they had to almost close their entire activity. And fortunately, this happened and they were able to start the work. And in the time where nobody could come out of the house, Fatima has gone from hospital to hospital and found out what is the requirement, what is it that they want, and they came up with the solution of the storage solution. Saliva collected from all over the places, transported without a cold chain, they could isolate DNA and establish the RT-PCR conditions. That is to the credit. Even now, their molecular solutions, their storage solutions are very, you heard one of the speakers say that they are buying it regularly from them, that these storage solutions work even at room temperature. there is no need for cold chain. That is a great achievement so far as in this situation in India, we need that very badly. I congratulate them for having found this solution. So, next slide, please. Uh, this is again talking about all the properties that were found and comparison with ethylene bromide and cyber green, and in every case it performed and as good as any other dye, and they even got the IP from 4 countries and everything. As I said, when it was just about to take off, then there was no money, there were no investors were coming forward. At that time, they, um, it is the mindset. If something is blocked, you turn some other way and find another path to move on, and that's what they did. Next slide. So then they are going to be using, now that they have established themselves with the micromolecular solutions and storage solutions, they are in a position now to be able to come back to the story of the dye and use it in many other molecular biology techniques. And therefore, I really wish what started in my lab, they will continue the research and be able to develop some of these, replace some of these dyes that are already in the market with the Azooka dye, the Tintor. I wish them all the very best. Thank you for your patient hearing. Anything else? I suppose there are no questions, so thank you all for your patient hearing again.
Thank you, ma'am. From Tintorag to today, your continued presence truly means a lot to us. Now, I'd like to call upon Dr. Usha to felicitate, ma'am. We will now go for a lunch break and we will assemble back in about an hour. The lunch is outside in the courtyard, so please. A serial entrepreneur and a molecular biologist, he has founded ventures like Enzene, Vipragen, and Genai, and has spent over 3 decades building and mentoring the biotech ecosystem in India. Can we please have him on stage? Next, we have Subhansham Baumik, who has begun his journey in the MBPU in Bangalore as a protein chemist before following his passion to found Bencos. Over the past 16 years, Bencos has catered to academia, clinics, and industry across India, South Asia, Europe, and the US, and post-COVID expanded into developing its own NGS library kits and clinical reporting solution. Sir, please come on stage. Lastly, we have Raj Wagre, a technology and entrepreneurship leader with over 30 years of global experience as the head of entrepreneurship cell at SID IISc. He has incubated more than 60 deep tech startups and actively mentors founders on scaling and fundraising. Our moderator for today is Josh Jacob Khalil, founder director of Future ICT Forum and India Advantage Summit, a strategic ecosystem builder focused on international collaboration, startup mentoring, and sustainability and digital transformation.
So I'll just make a small point here for, I think, thank you, Jose, for helping us to moderate this session. For all the entrepreneurs out there, please keep an eye on Jose Jacob. If you want to get connected to anywhere in Europe, be it Brussels, be it Paris, everybody's embassy or the consul general is on his board of directors. Okay, so he's also helping us get into WHO. So I encourage you to spend some time and get introduced to Jose Jacob. Thank you.
Thank you. Thank you so much, Alex. And nice to be here. You know, it's a very tough job, this job of being an entrepreneur, because you are always afraid of every step you take. You don't know which step might lead to a fall and which step might lead to growth. You know, the general statistics is out of every 10 startups that you know are established, usually within 2 years 8 of them close and only 2 continue after 2 years. Of course, in the newspaper we read the success stories of this, you know, small percentage of people who were able to survive and beat the odds. At the same time, many of the other stories are not there anywhere. And not that these stories are not important, each of them had their own personal battles, and therefore this session is very, very interesting and important, because with me are wonderful colleagues who basically help those entrepreneurs in their journey and help them manage their journey better and help them reduce risks, manage risks. I wouldn't say reduce risk, but I would say manage risks and create outcomes for a better world and better future. So thank you, wonderful panelists, and thank you, dear audience. It is post-lunch session, so we'll try to keep as much energy as possible so that all of you can, you know, enter the coffee break full of life and energy. So let me start with the first question. Maybe we start with the third speaker over there. The question is the same for each of you, and you can give your own individual perspectives from your experience. You know, usually when a scientist who has no business background, has no understanding of you know, debit and credit and, you know, balance sheet and assets and liabilities, you know, how can they equip themselves if they are deciding to take the plunge and enter the entrepreneurial landscape? So let's start from you, sir.
So I manage and run the incubation center at FSID IISc, and you know, I will talk in the context of the job that I do, right? So we have done about 150 startups so far, and most of them are about, you know, deep science, deep tech startups. Roughly 40% of my founders come from the university. They're faculty members, right? And this is a very unique challenge, right? Just the way you're saying, you know, people don't know about balance sheet and things like that. The challenge at the university is slightly different because they sit in the university and they write those big grants, and the grants come to them because of, you know, this great university, right? And sometimes some of them think because I'm able to get a lot of these grants, it should also translate to, you know, if I do a startup, should translate immediately to market success, right? And you know, that's really not so, right? And we all know that. So one of the things we do is, you know, there's a program by Gopal Krishna Deshpande Center at IIT Madras, which runs, you know, which has a 6 week, it's a 6-week program for scientists to understand whether their, you know, research is commercially viable or not, right? So it's a 6-week program. You start with the hypothesis, you know, go meet, you know, 20, 25 customers every week and validate, revalidate your hypothesis. And at the end of 6 weeks, you know whether, you know, there's really need for the product or not, or, you know, there's no need for the product, or there's, you know, a delta that needs to happen, right? For non-faculty members who come and approach us, we ask them to do pretty much the same thing in some way, form or fashion. Go meet the customers, validate the hypothesis, come back, right? So I'll stop at that and then.
Yeah, excellent. I think this is a very interesting thing that you have shared with us. This is a national level program, and people who come to you are able to apply to this program, right?
Yeah, so we nominate them to go and attend the program. This is for the faculty members at IISc.
Okay.
For those who are not from IISc, there's a different path.
There's a different path. Okay, perfect. Thank you so much. I think this is really important and interesting to note that there is a possibility to, do a test and pilot before the actual, jumping off, the cliff, let's say, and to see whether you will fly or whether you will fall. Very interesting. Thank you for sharing, that. We will keep that, scheme in mind. And of course, during coffee break, meet him to know more details as well. Yes, for you, from your background and in that context, how do you think scientists should go about?
Thank you so much. I always like talking about our journey because that's exactly what you're talking about. So right from research to business, no idea whatsoever of balance sheets, nothing at all. Just excited about an idea, just want to execute that for a very long time. So we started as a genomics company back in 2011. It's been 16 years now. So very few players at that point in time, and very soon we could see that we were a key differentiator in the market. Not many players, and science was our primary objective, and we were doing well. Over a period of few years, we saw our revenue growing year on year, but at the end of the day, cash in hand, nothing. So it was surprising that where's all the money going? So we thought maybe we need more working capital. Maybe we need funds. So we started approaching a lot of investors and talking to many people. And interestingly, similar age group, I found a guy who is a financial consultant. So he went through all our details and he said, hey, you don't need funds. You just need to fix your financials. So that's when we realized our entire pricing model was flawed. We designed it ourselves and we had no clue how to design the pricing. So they worked with us for 6 months, fixed things, and that's when the actual growth started. We got to realize that how important that aspect is, equivalent to what we are doing. And that is one of the reasons now we support a lot of startups with the sole objective because people come with excellent scientific ideas. And as Sir just mentioned, that one is, of course, how viable your idea is in terms of business. It can be a fantastic idea, but you cannot keep on working on it forever with no translation at all. So we do help startups in, kind of filtering those projects into, viabilities. And at the same time, whenever get an opportunity, I like to talk to several startups in terms of how you should plan your financials, your modeling of your entire financials, and how do you design your pricing. So I think it's a very important part of any entrepreneur's journey. We were lucky that we survived because we were growing, just the financials were flawed. But I think it's absolutely important to stay afloat. Absolutely, yeah.
Thank you so much for sharing your own personal story. Normally, there is a tendency for founders once they're successful to forget all the problems along the way and only remember all the things that work. But it's great to have you share this with us because I was also following some of the mentors in this space, and one of the, let's say, Jewish rabbis who talks about money says money is a spiritual thing, it's not physical. Even though some would say, in India when you do UPI, it's almost, you don't say anything physical move, but some Amitabh Bachchan's voice comes that money is received. So it's really important to manage it. Just like they would say music is spiritual. If you don't put the right notes together, you get a headache at the end, or you can have good music as well. So it's really important to manage the right note at the right time and the right allocation. Because if many startups are very excited with the initial traction and they grow, they can grow into a big debt if they don't manage their financials. So thank you for that insight. And of course, yes, some of you who are founders or no founders, meet him at coffee break to find out more details and go into that. So from your side, what have you seen from your ecosystem and also your personal journey, especially from the science to business, transition?
Thank you. Wonderful to be here, and also thank you, Alex and Fatima. The fact that we are here celebrating their journey itself talks kind of struggle they've gone through. And I mean, in biotech or deep science, surviving over a decade itself is a challenge and also is a success. I think just to come back to your question, that most of us, I think, come from a scientific background. They not worried about the finance and then how the company formed, run, sustained, and a whole lot of things, I think, which we understand later hard way. In my case also, I come from a small village, worked here, came to a startup company, and then IAC PhD, and I thought, a good experience to start a company. So I just left one fine day AstraZeneca job, and then I and then realized that it's not that easy. In fact, the only incubation center was SID those days. I did approach SID, and I didn't work out, and then I went out to set up my first company called Engine Biosciences. I think generally what happens is nowadays things are different. When I started 2 decades ago, the idea was that there were no funding available. There were no so much government support as is now. Also, the many incubators now, you have over 100 incubators. Those days there were no, there were hardly few. Idea is that you have an idea. Generally, biotech or deep science takes longer gestation time. So, idea is that you have an idea, but you got to sustain for a long time. So, I always followed 3 mantras in my entire entrepreneurial journey that I've learned over the time that you 3 months as one is, of course, how do you sustain, not just giving salaries and also managing a family, because it's very exciting to start. I think if somebody wants to start, I think that's the best thing to do. And once sustenance itself is not enough. I think the second mantra is do some work around while sustaining, some kind of transformational work which lead to some kind of IP and new direction or scalable business. And also doing these two, they try to find out if there are any leadership, like the way Azooka has came up with, pick up 1 or 2 areas or products or technologies where you can focus and become a leader. How you balance between these three is what actually makes a startup sustain, do something, transform yourself into the next generation company, and then become a leadership in that space. It's a struggle, it's like you have multiple ways of working on it. So I think probably we can discuss that little more details. But the struggle and the excitement go in hand in hand. I think major challenges are now they're getting funding at the right time. We discussed about it, and then managing the funding, finance, and making sure that we have a revenue model upfront, and then getting into the leadership role. I think these are the things I think is important for any startup to look at it. And most importantly, whatever you're doing, don't look at short term and see how whatever you concept you do, it may be very exciting today, but how you're going to scale up and make it as a product or service platform where you can kind of broadly expand your business. I think if you don't have that in upfront thinking, then you continue to struggle, but you sustain, but you will not be able to do some transformational and get into the leadership role.
Excellent. Very interesting also. Like you said, about 15 years ago, when you want, or even maybe 10 years ago, when you want to start up, there are no schemes, there are no funding. Even I started my company in 2011. So there was this board in Koramangala that said "to let." So when I went there, the guy didn't even want to tell me what's the rent, because he said you are too young. And then he said, who are you? How many people you have in your company? I was starting, so it was 3 people. He said, why do you need 1,500 square feet for 3 people? I said, we will hire later. So they didn't even reveal what is the rent. Then finally, it was interesting that he was asking, tell me who you know. So I knew some people that he knew. So I had to give their names and go. And then he said, I'll call you back. Luckily, those people said good things about me, so I got a call back. And the next day, he asked me, what rent would you like to pay? So, but that's a trick question, right? If I set too much high of a rent, he will know that I'm stupid. If I set too much low, he will know that I'm greedy. So, I was like, this is the market price in this area. I can't pay so much. I need a 30% discount. So sometimes it's really good to know what you want, because then you can put it out there and people can immediately understand and reach out and connect. Another thing that I want to share, also as a moderator is, some years ago, let's say 15 years ago, if you said you're a startup founder and if you're a man, let's say, none of the girls would, the parents would not consider you as a proposal, because they would say, he's doing something on his own. But if he was in AstraZeneca or in Philips, then it's a different story, right? Now, because of Shark Tank and, even in the village, the parents want their kids to be an entrepreneur, it's a little better. But, some years back, it's a big risk, especially you said about managing your own family and the company and the expenses of the company and your family. And it's not necessary that members in your family will reduce their goals because you are an entrepreneur, right? They will still want, and they meet other children in school. Your children meet other children. They all have different things that they want. So they will not say, "Yeah, you want to add?" Yeah?
Absolutely. My wife didn't speak to me for 2 months because I had not told her that I'm leaving AstraZeneca job. Anyway, she thought I'm gone mad completely. So I think it was like that. So
Yeah, I mean, it's a very crazy journey. So I think it's really important that luckily now, because of Professor having that special scheme to understand better, the decision making is much more structured than just impulse and sudden movement. So thank you for that wonderful first round and very honest and direct feedback. Even though it's recorded, I don't think it'll be public, but it's good for those who really want to know and take that plunge into entrepreneurship. My second journey is, once somebody decides to start, it's really important to find the right direction. Like today we see all the startups have AI enabled in their thing, whatever they make. Even if they make table, it will be AI enabled table, right? So, you ask them why, they would say, look, it's easier to get funding if the word AI is there in your startup. So I just want to, pick all of your brains on this particular thing. If some of the young people here decide that they want to do a startup, is there a particular field where India has an edge? So for example, if you say it's Mysore silk, there's no need to say much more. Nobody will ask further. Like if you say you are an IT company in Bangalore, they'll not ask, how it's possible, right? Because it's clear the edge is there. So what do you suggest the young people as also India's strength and also the ecosystem strength, so that they choose the right, how to say, sector, direction, area in the, let's say bioverse, that's the right word. I was about to say biosphere, but bioverse. And then how do you think they should or India as an ecosystem should plan so that India itself can become the top supplier in the world for that particular field? So maybe all of you have different answers, but let's hear from you again. You can use the same mic. Yeah.
So that's a very difficult question to answer. So for us, we get applications from multiple domains. We are domain agnostic incubators. So on any day, I could be, talking to founders who come from maybe say defense tech or agri-tech or biotech, clean tech. The domain for us really doesn't matter as long as, we as IAC are able to support them from a technology perspective. So two things are very important for us, right? One, the ability for a faculty member to mentor the startup so that, their access to the labs and the ecosystem at IISc becomes easy, right? And that is the only reason why you need to come to an incubator such as ours to get incubated, right? The second aspect is if you're able to find the right set of mentors to help mentor the startup. It's not possible in every domain, it's not possible in every case. Azooka, I think, has been very, very fortunate to have, great set of mentors out here, right? Uh, Again, if you look at it from a VC perspective, investors perspective, things keep changing every year. I've been in the ecosystem now for about 8 years, and I've seen, pretty much, multiple waves, right? Whether it is AI, whether it is, bio, whether it is clean tech, flavor of the month is defense tech today. So what is very important for us is whether, the founder is the right person building the product. The founder product fit has to be very, very important. If that happens, I think, everything else follows, right? It's a natural progression. It's also your ability to persevere and stay the course. Yeah. So success comes eventually, but, you need to persevere, stay, and move forward.
Excellent. So if you are someone who doesn't get upset in the Bangalore traffic and you have patience, that could be one trait that, that can predict if you have the ability to create a startup. Yeah, it's interesting to note that for ISC, making sure that the back end, chain is ready, only then you accept, because you don't want to also misguide this young person, coming into this ecosystem and not getting the right mentorship, because in the end it becomes a waste of national resource and time. So it's really good. I think that's a very wise path to go. So yes, some of the young people who are having their idea in stealth mode. What is stealth mode? The idea is in between your two ears, the left ear and right ear, not going outside. You can look at the IISc website and look at all the professors, the faculty page, and see who is an expert in what, to see if you find the right kind of people, and then, go and meet them, write to them, and that could be a good, I would say, shortcut and segue to build so that your proposal gets accepted. Perfect. Okay, perfect. So I think that's very good. Thank you so much for that insight. Now, sir, to you, what do you think?
Maybe I'll answer your question in two parts. So number one, which you talked about edge, Mysore silk, something which is edgy about India. So if you talk about our times when we started back in 2011, I don't think we had an edge. So all we had to do is just work around it, somehow get things done. And I remember many a times, especially for the international clients, they were pretty happy working with China because of low costs. The only way we could sneak in were, extremely difficult problem statements where they were stuck, and they just threw it at us as a challenge that if you can do it, the project is yours. And luckily we had an excellent team. We were able to, counter those problem statements, and we started, gaining projects in Singapore and Europe and so on. So that's how we created that. But entrepreneurs today, I think there is a lot of edge, because India is looked upon as, as a growing sector in all divisions. We are approached by several investors, several other companies, at an international forum. They see India as a manufacturing hub now. They look forward to a lot of opportunities for tech transfer, working around, how they can make it India and so on. So I think there is a lot of edge and opportunity today. If you come up with, some nice innovation, something that caters to a global problem, I think there is a lot of scope and opportunities. Second, for startups today, I think the primary thing that the founder needs to do is, apart from his instincts and the ideas, the one thing that one needs to work upon is that, is my model solving a problem? There has to be a problem, and I'm developing a solution to it. And then the second thing is the problem localized or is global? So an understanding of that itself gives a lot of push. And once you are clear about that, and then neither time is a factor, neither funding is a factor, everything falls in place. So once you're very clear about that you're solving a particular problem, I think that's where the excitement and the zeal comes through. And if you have a fantastic team, you can definitely push it through. And of course, you need to have good financial understanding to plan your finances.
Yeah, excellent. You see how the second answer is feeding from the first answer itself. But the key I hear there is, search for problems, and if there is a problem that you are resolving, the chances are much higher of success. The key thing is to have the right team and make sure that the organization set up, the culture is based on innovation, so people can innovate together and build together. So, really critical. But overall, I think what I sense is India is also a market and a marketplace. Yes, please. Yeah.
I'll just add one last thing, the last part which you asked, that how can India become a global power in this space, say research and healthcare. So in my entire journey all these years, of course, when I started my journey, I also wanted to change the world, right? But then reality dawns in and then you slowly realize how things work. But if I see one thing as a major challenge, that is, I think at an educational level, the way we are trained in India. So I think somewhere, I won't say entrepreneurship, but the thought process of taking up a challenge and solving it among students, I think that has to be inculcated. That is something which is missing. Even if you do a lot of entrepreneurship programs, you talk to people, they come up with an idea, they start it. But again, if you see, as you said, I mean, let's start something in AI because it gets money, right? But we are not thinking about coming up with an innovation that stands out globally. To be a global leader, I mean, I always say, I mean, some may like or doesn't like it, but I call it as an Elon Musk way, as a Musk way. Something that people are not even thinking of. If you can think about it, there's always a solution that can come up. So I think that mindset is something that we need to work on. I mean, be it incubators or be it us, whenever we get to interact with entrepreneurs, I think that particular thing should stand out. That think out of the box.
Excellent. Indeed, it all starts and ends, with the mind. So if some of you were in the morning presentation and heard about, getting migraine headache, then, the problem is in the gut. So, there are so many problems to solve and so many unique ways to solve them. And I think it's critical to, especially for India, given that we have a huge and large market and biotech as a space is going to expand, it's really important to find the hard problems and solve it with a good team. Okay, sir, what about you? Your thoughts?
I think I agree with the co-panelists that you got to solve a problem to think about it, what is that you're going to do. At the same time, I just give my kind of real-time experience starting 3 companies, and the first company started as with my own experience in the recombinant DNA platform. That's the only thing I knew. I always wanted to be an entrepreneur, just jumped out from the job and started that. So only thing I knew was clone and express make proteins. So that time I was very clear that there was a lot of pharmaceutical company were getting into biologic space. We knew that there's a huge space by biopharma space. So the first thing I did to sustain we did everything, made products, services, sustained, and also never let go on setting up some kind of world-class technology platform to set up the biosimilar development. So that's where we started working with pharma company, worked with Alchem almost 10 years, developed almost 7-8 molecules. And then Alchem has bought my company. Of course, I exited very early, now inching towards $1 billion company. I think this kind of gave me some kind of experience that I think many people told me that, first company was a fluke. It just happened like that. And then I started a second company, which was again, as you said, there was a very important requirement for a country like India to have some kind of drug discovery driven contract research organization. So we went to the Mysore and we floated this company along with a couple of scientists. Now we are around 100 people trying to work with, hand-holding a lot of innovators. We are becoming part of solving their problems and also helping their research to take into a commercial level. The third company which I bought a small business from Bangalore, Jenny, which was sold to Merck. Bought that business back and ran it successfully for 4 years and exited. I think there is no one single, recipe or way of starting something. I think you wanted to do something and it just gets started. I think you need to put together those things once you have this. The fourth company which we have just floated along with some of my very fine scientists, uh, again trying to understand that 90% of Indians can't afford biologics in India. Now we've discovered that some of the technologies now, both in the cell engineering as well as in the upstream platform, we will be able to solve this problem to a great extent of taking at least this 5% affordability to 10 to 25% if we do everything in right. It's a startup, but we are looking at long way. I think it is again working with partners, collaborating it, and both in terms of finance and business, and also the technologies. Some of the technologies which we are working with our new companies, we are bringing a couple of cell engineering platform which are giving almost 10 grams per liter. So we are signing up with them and bringing those technologies to India and make it make in India for India kind of things. I think looking at that kind of long-term vision, you don't start something to make some money or something. Money, I think for us, making money is only if it happens, it happens. Otherwise, it may not happen end of the day. I think that excitement of starting something at the same time thinking long term, and especially what is that impact you're going to create on the people and the ecosystem, also on this society at large, is actually what makes an entrepreneur every day get up. Sometimes you don't even sleep, and then the days go on.
No, indeed, there is this philosophical note that some people get up in the morning and say, "What do I need?" These are called the needy people, and some others get up, which are a few percentage. Hopefully, in India, that percentage goes up. Who gets up and says, "Who needs me?" And this person goes out to solve whatever can be solved. I think in India, given that our demographics and a lot of people are young and also ambitious, it's a very good time to solve. And you have across the different quadrants, affordability, quality, time to market, speed, innovation. Everywhere there are challenges, and you can solve across. At the summit that I run, which is called the India Advantage Summit, we have seen now drone companies that want to transport organs, because otherwise the traffic is too long and they get stuck. We have seen companies that are trying to solve for energy. They're trying to solve. So there are a lot of young people who basically look at an impossible problem and jump in and do their best. Well, this gives me the segue to our next very critical question, because while everybody talks about the successful few and success, very few people, especially in India, like to acknowledge failure. And unfortunately, or fortunately, because I have a company, I know that failure is as important as success, because every failure can teach a lot of lessons in itself. So I want to ask each of you, what is your advice, especially when scientist fails in the with his starting with the hypothesis and goes on and does all the things, and there it's the grant based model, right? Already the money is there. In the end he says, okay, there is no positive correlation from this to that. But when he's doing a product and then failure comes, there are real consequences. You may have to fire some people in your company, you may have to move to a smaller office, you may have to downsize, downscale, and and some of them shut down and then maybe restart or sell the company. So just give us some example stories or something that you heard in your years of experience or you personally experienced, so that can get some lessons of resilience for young people who want to start. Okay, we can start from this side then. Yeah, you can start and then he will close in the end. By then you think of some story that comes to your mind.
There are many examples. So I think I made more mistakes than right, but I think one right changes the whole game for you. So just as an example, So during COVID, I think that's when everything was shut down and things were all stalled. Nothing was moving. Most of the projected revenues suddenly, disappeared by the last quarter because from February the lockdown, initiation started and everything fell apart. But I think one very important thing, since most of us here come from a research background, I think one very important thing that has helped always is that whenever we run an experiment and it fails, we know we can troubleshoot it. So that mindset has helped a lot. So I think the first thing that one needs to always remember is not to be too obsessed with what you want to do or the idea that you're carrying. Always be open to troubleshooting. If you face blocks, stay open to keeping yourself afloat because that's more important. If you survive, your venture survives. So you have to be open and flexible in that part. And I would say that we have been lucky, in an opportunistic way, you may call it. We survived COVID. We, got into something which got us funds. That little period 2021, and then we were back on track, and then the growth continued. So I think that mentality matters a lot. That instead of just being, kind of depressed about, I've lost it or it didn't happen, just sit aside, troubleshoot what can be done, and if you need to take a sideline, because I believe in an entrepreneur's journey, nothing is right or wrong. At the end of the, if you reach your, end line, I think that's what matters.
Excellent. So, that's very good. I would put it that, important to be willing to pivot and flexible. So here again, the concept of humility, because, if you are humble, then you can see the signals. If you are very proud, then, that's also linked to my next question, but I think we'll cover it here, that you can get depressed because you feel the weight of expectation of other people on your shoulders. So it's really important to be a light soul who is not heavily burdened, who is humble, and who can make a joke and say, hey, we were doing that, but now it's difficult, it's not selling, so we're doing this. So that's really key, to be flexible. So while you're students and studying and doing your research, try to see how you could develop these qualities, and pay more attention to troubleshooting and join those teams that do troubleshooting. Yes, sir. Yeah.
No, I think failure is kind of, never know how, when you're going to fail in the entrepreneurship journey. You don't start something, a futuristic deep science company, thinking that I'm surely going to succeed. I think that's not generally the case. I think we, again, when you start a journey, many times when I'm in my first company, even after 3 years, we were struggling to pay my son's fees. My wife told, go back to a job because I was the only source of income. So I only told my wife that, look, I only take one way. So either I will end this, otherwise I'll go back to my village and do agriculture. So it was like that commitment that, you want to go forward. You don't want to come back, whatever happens, come what may. I think will actually make you think, find alternatives, they figure out, find a partner. You never believe, I even taken money from my guide in IAC. He has invested in my company. So, and when I given him back 4 times, and he said, did you, loot some bank or something like that? He told. So it is like, it's like struggle at the same time. When you succeed, you have that kind of feeling. For example, when you start a company, whether it is 10 people, 5 people, now we have in Mysore, I run a company called 100 people. We have 100 people. It's not about your life, your family. So you are basically taking care of making sure that they get salaries every month. So your responsibility is so much. There is no way you can go back and then, these days I'm not going to, I will just close my company. That doesn't happen in reality. So it's not that I've started a company and then I close it down today and tomorrow I'll go and join a job. So that doesn't happen. So it comes with lots of commitment and responsibility in addition to, running the show or whatsoever.
Excellent. So that's a very wonderful way to end this panel, because we are trying to catch up time and be on time. Commitment and responsibility should be the core guiding principle, and I think then your venture has all chances to succeed. You want to add? Yes, yes, please, please.
I just wanted to add one thing. There's no shame in failing, right? I mean, you fail, you fail. You shut down shop, go do something else. I did that. My business didn't run. I came here, I took up a job, and I'm working. Right? So
Excellent.
life is much bigger than just that small.
Yeah. So to have an open mind, let's give a big round of applause to this wonderful gentlemen who have shared their stories, and handing over back to the organizer. Yeah.
Thank you to our moderator and our panelists. It was truly an engaging discussion. It was truly an engaging discussion of what it takes to build and grow. I would like to now call upon Dr. Ashok Gopinath to felicitate our panelists and moderator.
So Ashok happens to be my batchmate from my IIC. Wonderful to have him and receive this from him. Thank you.
It is my pleasure to introduce our next speaker, Dr. Major Satish, the founder of AI Health Highway and an AI-first medtech company, transforming how chronic diseases are screened at the primary care level. Since 2018, he's been on a mission to make early detection smarter, more affordable, and accessible, aligning his work with vision of ensuring healthy lives for all by 2030. With over 20 years in healthcare, an MBA from IIM Bangalore, and training from Stanford, he brings together clinical insight, strategy, and innovation in a powerful way. Dr. Satish, the floor is yours.
Good afternoon, ladies and gentlemen. I think after lunch, it's always a tough task to hold everybody's attention. I'll try to do my best. So it's been a long journey. I don't do clinical practice, but a brief background. Most of my practice was with the Indian Army, and I'm very thankful to have got an opportunity to serve the country. So currently, I lead the startup AI Health Highway. We started this in 2018, and been a very exciting journey, I should say. So why AI Stealth? I should point it. So I'll just talk about the statistics, why we started this company. If you look at the world's biggest disease burden, it is non-communicable disorders. And every year we lose about 43 million patients across the globe due to non-communicable disorders. And within this, the cardiac and respiratory disease segments are the major contributors. Also, as an Indian, why it is important to solve, because most of these deaths happen in the age group of 15 to 60, which is economically the most productive age group. So when this happens, it's not just that individual, but it takes the entire family down the poverty line. So with that background, we started healthcare. It's a fundamentally flawed concept. When we say healthcare, we think about hospitals. I consider hospitals as a garage. When an accident happens, you take a car to the garage. But actually, healthcare starts much before that. If you do a good job with primary and preventive healthcare, if you take care of your car, you do the servicing well, you don't need hospitals, or at least you can delay. So it's very sad to know that in 2026, with all the talk of AI, generative AI, LLM, and all this technology, whatever has been happening, still 50% of the world's population does not have access to essential primary healthcare services. Forget about hospital. I'm talking about essential primary healthcare services. So that's where we didn't want to build one more device which would go into the hospital. Even today, 60 to 70% of India's population has to travel 50 to 70 kilometers to reach a hospital or to see a specialist. That's how we went back to the drawing board, and then we came up with AI Steth, because a stethoscope is probably the most widely used tool. It was invented almost 200 years back. So our hypothesis was, if we are able to innovate around the stethoscope, it's already there with the doctors, the reach is there, then we would be able to solve majority of at least the cardiorespiratory disease segment. Now, why it is important for a country like India? 1.4 billion population, we just have about 6,000 cardiologists, 3,000 pulmonologists. But India is a country of 6 to 7 lakh villages. You won't get to see someone like Dr. Devi Shetty, Naresh Rehan, Dr. Naresh Rehan in villages. So our idea was, if we are able to bring that expertise through technology, then we can take this to all the 6 to 7 lakh villages. And how this idea was born, all Indians follow cricket. So till 2012, the caught behind decision was taken by umpires on the ground. I consider them as cardiologists or experts. But then came a device called sneakometer. It helps identify whether the ball touched the bat or the pad. So then it's not just those two umpires. Even whether you are in the stadium or you are watching the cricket match from any part of the world, you can decide better than the umpires whether the ball touched the bat or pad with the signatures. So we used a similar technology to study the heart and lung sound signature patterns. So that is how our journey started. And I'm not sure if the animation plays, but essentially AI Steth is a platform which has got three key components. The device collects 30 seconds of heart sounds, 60 seconds of lung sounds. It is streamed via Bluetooth to a smartphone. And once this gets uploaded to our cloud, our AI engine analyzes the signature pattern and gives a result whether there is a presence or absence of murmur. Why this is important? I'm a doctor, and if there are other doctors in the room, when I make this statement, it's a huge ego hit for them. Who are the doctors in primary healthcare clinics? Most of these are MBBS doctors who have just graduated. So the accuracy of a newly trained doctor when they use a traditional stethoscope is 50 to 60%, which means 1 in 2 cases missed. When the AI studies these patterns, we did our first pilot study in Jaideva Institute of Cardiology, where the murmur detection accuracy or sensitivity is 98.34% and the specificity is 99.73%. I'm very proud to say that now this is deployed in Maharashtra, where a ASHA worker uses this, takes this door to door, and in 30 to 60 seconds does the initial screening. How this impacts public health? Earlier, if 10,000 villagers had to be referred to Pune or Mumbai for echocardiograph, currently the gold standard test is echocardiograph for valvular heart disorders. It takes 30 to 45 minutes. It costs around 1200 to 4000 rupees. So now we are able to screen at a primary healthcare clinic in a village, and out of those 10,000 villages, only the 1000 or 2000 who have this murmur are referred to the cities. So this is not just triage and screening. Think of all these people. When we go to, let's say, places like Gulbarga, Raichur, some of our projects are running in Uttar Pradesh. These are all villages, daily wage workers. They have to come to the city. By the time they meet a cardiologist, it is 3, 4 p.m. in the evening. They'll get the report next day. So it's not just a test, but they lose their daily wage for 2 to 3 days as well. So this is how we started. In the last 18 to 24 months, we have screened about 70,000 patients across 20 states of India. Maharashtra has deployed this for adult valvular heart disorder screening as part of a national program. And Uttar Pradesh just launched this for screening children who have hole in the heart. So India produces 3 lakh babies every year who are born with the congenital heart disorders. And out of these, at least 2/3 need a surgical procedure. We are able to do only about 25-30,000 every day. So with this screening and detection, Uttar Pradesh launched, along with Stanford, a 10-year initiative to eliminate rheumatic heart disease in the next 10 years. So what we know more or is in the public limelight is about the acute heart attack. But the next big silent epidemic which is coming is the valvular heart disorders. This is more chronic in nature. For a country like India, one segment is the rheumatic heart disease or children who have hole in the heart. But the second bigger disease segment is secondary to hypertension. Once you have BP, the wear and tear of the valve progresses for 10 to 15 years. And this is something which is going to increase in the next 10 to 15 years. And we are working in both these segments. So our idea was what used to be the expertise of a cardiologist, pulmonologist, how we can take it to each and every village through AI STEP. So while we started with cardiology or valvular heart disorders, we got feedback from the MD of National Health Mission in Maharashtra. If in 30 to 60 seconds this device can be used, someone like a ASHA worker or a MBBS, why can't you also look at the respiratory? So now currently our work is under clinical validation for respiratory disease segment. So in a fully developed model, AI STEP will be able to screen, triage and impact 6 public health diseases: valvular heart disorders, heart failure, COPD, asthma, tuberculosis and childhood pneumonia, all by using a simple device like a stethoscope. So just want to highlight, I think there was a lot of talk about how a founder's journey when we started. Into 2018, when I pitched this in India, nobody believed. There was no talk about AIML or whatever. So we first got selected to an accelerator in the US. Then the early prototype and product was built with Sound Hub in Denmark. Then we got incubated in FSID, Indian Institute of Science. And thanks to the excellent team, Raj, Natras sir is there, Murli sir. So we work with Professor Prashanta in IISc. And I'm very proud to say after 6 to 7 years of hard work, just in Jan and Feb, we got 3 patents for this product in US, Europe and India, all 3 regions. So what is the fundamental shift in technology? If you go to the previous slide, you have to understand how does a human being learn. So 30 to 35% of a population is acoustic learner, but majority 60-65% is visual learners. So that's one of the reason if you go to a doctor and 3 doctors see you, all 3 might have a different opinion. But now what you might miss hearing, this phonocardiogram printout captures and marks if the murmur is heard, is it in systole, diastole. While we started with that segment, now we are moving into even identifying whether the murmur could be due to aortic stenosis or is it due to mitral regurgitation. So then we discovered multiple clinical segments in this journey. So these are the study results from our Jaideva. Now ICMR is doing a multi-center trial. So we have about 4 publications in IEEE and recently got 3 patents. Sure. So we can skip this slide. These are all specifics about our AI engine and so on. So if you just go back to the previous slide, this is the current work that we are doing in the lung sounds. Currently, it has an overall accuracy of 76% for identifying whether it is a normal or abnormal lung sound. So now we are going into even identifying whether the patient has COPD, asthma, tuberculosis, or childhood pneumonia. I'll just spend 30 seconds to explain why this has much bigger impact than cardiology. In most rural villages or places where you don't have chest x-ray, a doctor has to take a decision if a child has pneumonia on the 3rd day whether to start antibiotic or not. Otherwise we might lose him as well. So when we learned medicine 25 years ago, it was completely a different world. There were 3 fundamental steps. 50% of the diagnosis came from the patient's history. Then you did a general physical examination, you used a stethoscope, and you arrived at your differential diagnosis. You were relying on whether it's a chest x-ray, CT scan, only to confirm the diagnosis. But now it has flipped. I don't know how many doctors would have time to listen to the patient's history. As soon as you go, you are given a set of investigations to do an ultrasound, CT, and so on. So that is how healthcare becomes more costly. So we went back to the fundamentals and then looked at how we can again reinvent the fundamental tool. So these are our product portfolio. Maybe we can skip this. Yeah, and I think I already spoke about these. These are some of our ways how we are disrupting through technology and innovation. We can skip this. We can skip this as well. So these are all are different. So one of the fundamental challenges in the first 4 to 5 years, this solution was available only where there is Wi-Fi or internet. Last year, October, November, we got selected to Google for AI first cohort. And then we cracked now, there is a solution or MVP where this can work when there is no internet. Most of India still does not have internet access. So this will be available even offline. So I think we can skip the next couple of slides. These are all, so this is like a dashboard of our work. We can skip this. Yeah. So this is our team. As I said, like we work closely with Professor Prashanta and our early journey was with SoundHub in Denmark. Right now still a very lean team of about 18, 20 people. Yeah. So these are all our rural screening camps that we do. Most of the deployments are in public health clinics. I was very fortunate to be called by Mr. Ratan Tata. He personally called and gave a feedback about this product. His input was why you're building this for doctors. Maybe as a patient, I can use it myself. Maybe someday in future, like how 20 years back, even to check your BP, you had to go to a doctor, but now you can do that yourself. So in the larger scheme of things, as we do more screening for chronic diseases like asthma, heart failure, our product would be probably someday used by patients themselves as well. Sure. So these are some of our awards and achievements. We are backed by Rainmatter, TurboStart, ISC, Social Alpha, Tata Trust. So thank you so much. And I would like to congratulate Fatima and Dr. Alex. Congrats on your 10th anniversary and thank you for giving us this opportunity. Really appreciate it.
That was a great reminder that AI isn't just about innovation, but also accessibility in rural areas, sir. Thank you, Doctor. Now I call upon Jose Jacob, sir, for the felicitation of Dr. Satish. And for our final speaker, today we have Dr. Ashok Gopinath, a genomics scientist whose work beautifully bridges deep science and real-world impact. With a PhD from the Indian Institute of Science and postdoctoral research at Cornell University, his early career focused on using genomics to answer fundamental questions in biology. He then carried that expertise into biotech and pharma, including a significant stint at Sanofi, where he worked on applying genomic technologies in areas like metabolic disorders and oncology. From research to boardrooms and now leadership roles in Illumina and Strand Life Sciences, his mission has remained consistent to move genomics beyond theory and into clinics and ultimately into the lives of everyday people. Please welcome Dr. Ashok Gopinath.
Thank you. Am I audible? Yes? Great. Could I have a pointer? Give it to me, give it to me. Which is the pointer? Forward, reverse, pointer. Yeah, so thanks for the elaborate introduction. I currently work in a company called Strand Life Sciences. Many of you may be aware. It's a bit of a homecoming for me because Strand Life Sciences 26 years ago was started by 4 professors from IISc, from the computer science department. I think some of our esteemed guests here today would know Dr. Chandru and Dr. Ramesh and others as well. If nothing else, we'd all meet at the same tea board or coffee board at some point of time. But the important thing is that life has different journeys planned for us, and I'll take you through mine. The only unfortunate thing is I date myself and you'll begin to understand how old I really am. I try to get the best picture of myself here so that you understand that this differential is always too high, right? But yes, some of the things have already been mentioned. I did a PhD in IISc in the Department of Biochemistry. Had some stints in Sanofi, spending time with oncology. Was in a small startup called Connexius, where I did systems biology. And of course, I went on to head Illumina in India for about 5 years in between. And all of these have been extremely wonderful learning curves. And I've tried to intertwine that with some of the progresses that were made in biology. The only disclaimer I have is I didn't make all of those discoveries. It just so happened that they happened when I was doing my PhD and making my way forward. Just to orient everybody here, I know maybe not required for this audience, but genomics is that study of the various cellular interactions or transactions that take place that involve the genome, central to which is the DNA molecule. And the most primary layer at which DNA is identified is at the level of sequencing, where the order in which the A, G, T, and Cs are established for posterity. This molecule has been something that many of us have worked with, published papers on and stuff like that. But primarily its utility in the world today, the consumer world today that I work in, goes back to something called the central dogma of molecular biology, which is that this blueprint, this molecular blueprint that each of your cells has of DNA is believed to transact into a intermediary molecule called RNA, and then of course that into protein. And it's these proteins, there are various kinds, structural, transport, regulatory, so on and so forth, that then go up to make that cell into this living, thriving, functioning unit that we are all made up of, right? So it becomes really important then as an extrapolation to say that if there is something going wrong there, which we will call pathology, then if we trace it back, we should eventually be able to find it here, right? And so if we simply sequence the DNA and we find errors of the organization of the DNA here, then are we able to predict pathologies that would manifest at the other end of the spectrum? So the goal is simple. If we sequence your DNA and you haven't yet got any pathology, we'll be able to find out if you will get one. And if we do find that out, then what are the ways in which you can intervene and prevent that from happening? Okay, so that's the whole goal of wanting to understand what one's DNA sequence is. again, it sort of manifests in the form of disease prevention per se. So India as a country, I think during the 1940s when we got freedom from the British, the average age that one would, life expectancy was 37 years old, right? And today it's in the 70s, right? There was a small blip during COVID, but for the most part, it's in the 70s. So therefore, a lot has probably happened in order to make that something that we can be proud of today. So one of the things was obviously vaccination and infectious diseases, and of course antibiotics, and a lot of public health programs on sanitation, public sanitation, and those kinds of things, right? That sort of conspired together to actually improve the, excuse me, the life expectancy that one has in India. I'll just go back. So there is a very philosophical question as the title of this slide, what is life? But the answer is very science answer, right? It's that span of time that you have between birth and death, right? I won't get into the rest of the philosophies, we can discuss that later, right? But anything that happens between the time you were born and the time you die is what we're going to call life. And it would be very useful to know that if we can predict things that are going to happen to you later in life early enough, can you prevent them? Can you delay them? And therefore, can you live longer? If you were to take the numbers from 2019, there were 55 million deaths recorded in the year 2019. 74% of those deaths were from non-communicable diseases or chronic diseases, right? You can see infectious diseases and newborn diseases are 2% and 3%, 14% and 3%. And this goes to show you that tomorrow's health problems and health pathologies are not going to be from having, solutions may not come from having antibiotics. There's going to be a bigger, more all-encompassing approach to this, because non-communicable diseases are going to be what kill most of us. So there is this same sort of cycle of life that I had described earlier, right? If one were to sample somebody at the age of 50, which is that guy with the briefcase, maybe somewhere in between that guy with the briefcase and the slightly overweight guy. If a symptom appeared there, that's roughly around my age, around in their 50s. Today, what people would do is they take therapeutics for this, right? So I take a metformin or something else, some anti-obesity drug, and of course try and work my way and extend my life till as long as it can be. The goal of using DNA testing at scale, right, that means everyone in this room would do it. The goal of doing something like that would be to identify early enough if you have predispositions towards getting a disease, right? Making modifications via wellness and lifestyle, and as a result of which, maybe getting that little expansion, lengthening in your lifespan. And of course, it helps us define something called the health span, right? So you can stay healthier for longer, and therefore you live longer. So these non-communicable diseases are generally caused by two major determinants, right? You have your nature and you have your nurture. So all the studies that we do with DNA are addressing nature, right? This is something that you're born with. And of course, the kind of screening that I'm going to talk a bit about to you today is genetic screening and what it would mean. But of course, what we're all very familiar with is the role that nurture plays in our health. For example, that particular gentleman having a soda and smoking a cigarette is probably going to have a slightly shorter lifespan than the rest, for those who don't have it, right? So if we can address both these determinants, do we set ourselves up to have interventions that can have us have a longer life? In addition to these kind of both ends of the spectrum, right? There is a group of genes that are affected by an interaction of your environment with your genes, right? And some of those genes are genes that we recently characterized and put on a panel and now is available for people to test themselves on, because they very conveniently help you decide whether you are more prone to have some kind of micronutrient deficiency or not, and then you can take the necessary action. So Strand as a company, is one of its goals is to take genomics to everybody, right? And the reason to take genomics to everybody is to give everybody a heads up on where their health is and what they were born with as maybe variants that require them to have a closer look at their, either their sleeping pattern or what they eat. And it starts with a cheek swab and ends in 14 days with a report on one's phone. The product is called NutriDNA. It's basically a report that is divided into 8 different categories, right? Vitamin and mineral metabolism, food reactions. There's a segment on optimizing weight loss and long-term management, weight management. There's a little segment on eating behaviors, some factors that influence metabolic health. There are a bunch of genes associated with exercise and maybe injuries that you might have. And then, of course, inflammation risk. And finally, we have sleep quality and lifestyle. And all of these are genes that are assessed. And then, of course, characterized. And then some level of recommendation and intervention is given back in this report. And this recommendation does not include supplements, right? So it's invariably some kind of food that you might eat or something like that, right? Or an exercise activity that you can undertake. So one such example I've taken out of all the genes that we look at is a gene called the FTO gene, right? And this gene has a known association with actually impacting appetite. So people who have this particular T2A variant in their FTO gene are not known to have a lower satiety signal from eating. And such people, through observational studies, have been documented to be, on average, heavier, right? So obesity is something that they're more prone to have. So if you did such a test in advance and you found out that you have this particular variant, then the simple solution may be as simple as having portioned meals, ensuring in advance that you don't overeat, because you can't trust your body signal to say when to stop, right? There are other aspects to this. For example, if your diet has more protein in it, then you can eat just as much as you want to eat, but it doesn't impact your weight gain, right? So there are many sort of clinical trials that have also been conducted in interventional studies using this for this particular variant in the FTO gene, and well-studied examples of how changes in diet can actually help you maintain your weight. Another example that I'm going to talk to you about is in the optimizing exercise and fitness segment. So there's a gene called ACTN3 or actinin-3. It's also called the speed gene, something that is present is actually well expressed in athletes. So people who have this high burst speeds of energy, sprinters, they all have this. It's a component of the fast twitch muscle fiber. I'll give you an example of myself. I have a stop codon in this gene, and so I'll never be that 100-meter sprinter that I wanted to be, okay? But that being said, it also helps understand whether I will have an adverse impact on my muscles while training. And so just because I'm not going to be a sprinter doesn't mean I don't try to sprint, but the chance that I'll get hurt is also pretty high because this gene is not present. And therefore, if someone were to recommend a fitness program for me, the goal would be to have slightly more cardiorespiratory types of exercises as opposed to a whole bunch of high sprint, high performance exercises. And one last example I'm going to give is I think in vitamin and mineral metabolism. And the reason why I give this is because this was a very useful thing, particularly for me. So, as a 50-year-old, when I was designing this test in Strand or putting these set of genes together, I hadn't done a blood test for 20 years. I had no symptoms of, the least of which was a vitamin B12 deficiency symptom, which, by the way, for those who didn't know, is foggy brain and tingling extremities. I never consciously ever thought I had that, and so I just never did a blood test. But while I was building this genetic test, I realized that I actually have a high risk because this FUT2 gene, had a stop codon where it shouldn't have had one, right? And that is known to, and because it's a non-functioning gene, there is this protein in the stomach that chaperones vitamin B12 from the stomach into the intestine before it can be absorbed, called intrinsic factor. And because it's not modified, the intrinsic factor doesn't do its job, and so you have a huge problem. People who have this mutation have a problem absorbing vitamin B12. Now, my first thing to do was to do a blood test and see if I was actually my vitamin B12 deficient, and lo and behold, I was, right? And I'll give you some of those, those graphs. But just to give you an idea before that is what really happens, and this 52-year-old male is me, so I have the right to talk about this as an anecdotal thing, but I see many people since who've got the similar sort of readout. So I was an asymptomatic 52-year-old male who's non-vegetarian. By the way, B12 is present in non-vegetarian food and I shouldn't have been a candidate to be B12 deficient. But my genetic test predicted I'd have low vitamin B12 absorbance. So when I did my blood test, sure enough, I had low vitamin B12. Now, I'm in the presence of maybe the person who knows this pathway the best, the SHMT pathway the best out here, right? And so she's most familiar with it, Dr. Savitri, I'm sure. But for the rest of you, I'm going to sort of reduce this pathway down to, if you have low levels of vitamin B12, you are very likely to have a collection of homocysteine in your blood. Okay, it's going to get trapped there because B12 is required in that enzyme methionine synthase to make methionine, and if B12 is low, that pathway of homocysteine going into methionine is blocked or appears blocked, and so your homocysteine levels go super high. Now, this is a test that not many blood diagnostic centers will actually offer you, but you can test your blood for homocysteine. Okay, so the natural thing for the scientist in me to do was low B12, prediction is high homocysteine. Let me see. Before I tell you what that result is, just to build the suspense, having high homocysteine is not a good thing. Okay, homocysteine is one of those amino acids that is known to actually shard through endothelial cell walls, right, and cause injuries to these arterial walls, accumulation of fatty substances, attraction of an immune reaction over there, and of course, in the end, the resulting result is in clots. These clots can go into your heart and cause a cardiac arrest. So someone with high levels of homocysteine, just FYI, anything up is considered high for homocysteine. So it's not a good thing to have high homocysteine. So B12 is low, homocysteine is high. Let's see how I turned out. So we'll start on day 0. My homocysteine levels were closer to 30, okay, which is not good for me. Now, mind you, I'm an asymptomatic individual just designing a genetic test before I discover that I'm actually possibly a walking heart attack, right? But now that I know I have homocysteine levels this high, my first, easiest, simplest thing to do was to take oral tablets, supplements, B12 supplements. But if you remember my slide earlier, my absorption of B12 is what is impaired, not my access to B12, right? So given that my absorption to B12 is what's impaired, it's unlikely that taking oral B12 would have made any difference to me, and that's kind of panned out as well, right? I may have had a marginal decrease in homocysteine levels because I was overdoing on the supplements. So I did an intramuscular injection once a month for 3 months of B12, and my homocysteine on the last column there has come well under control. So I'm a standing example of how when you think you have no problems in life, right? Until someone comes up with an irritating DNA test, asks you to do it, you realize that you have something and you can change it just by taking an intramuscular injection, right? So whatever risks I bore of having a cardiac arrest from homocysteine levels increasing has been mitigated, right? And so basis this example, I mean, we look at various other kinds of, we have about 55 different traits that we actually, we've compiled together that will give you some idea of what your genetic risk to getting a particular type of either deficiency or inability is characterized. And then of course, there are recommendations that we give in there. More recently, whenever you deal with consumer markets, right? Whenever we were talking, I think my title says something like, increasing the access to genomic solutions. This is ideal as a tool to increase access to genomic solutions because you can actually help yourself, right? And one of the things that many of us in the consumer space face is sharp criticism almost all the time, right? So one of the things was, hey, hold on. This slide deck has a life of its own. Please bear with me. Can we go back? Back again, please. Once more. That's fine. Yeah. So one of the criticisms we got was, okay, so you tell us what our problem is, but you don't tell us what our solution should be, right? And so one of the things we incorporated into this was an AI meal plan. So based on your genotype, we have an LLM that runs in the background that basically compiles all your genetic risks and then assumes that you have certain goals that you have to build. Either it's weight loss or weight gain, or you want to stay fit, whatever it is. You put these parameters into it and it generates a weekly meal plan for you. So then you can adjust your diet to what your genes kind of predict you could do with in terms of supplementation. We have one more product that we have, and that first product was something that we did more or less in reaction to what I think consumers kind of gave feedback to us on. We started out with the product I'm about to describe to you, which is learning about the nature more deeply than these sort of in-between genes. Right, so this product that we brought out is also a genetic panel, also starts with a cheek swab. And you can see whenever I mention the word cheek swab and those kinds of things, there are, there are many reasons in which, we use tubes and other such things, and you're in the presence of places that know how to do that the best. We take that part of it for granted. One of the tests we do, for example, Genomic Health Insights, starts with a cheek swab. This is a far more serious test, right? We understand, for example, what your predisposition to having hereditary cancer risk is, or maybe what your risk for hereditary cardiovascular diseases. We also do something called carrier risk screening, and then there's a segment in there called pharmacogenomics, where we actually try and understand and help you understand how your body metabolizes drugs, so that you can then optimize either your dose or the type of drug that you might actually use. We started out by rolling out this product. And as high as the value proposition for this product was for us as scientists and human beings who mean well for people, people used to be really scared to do this test. Because the consequence of finding something positive in this test means a clinical intervention is required, which means you have to go to a hospital and your doctor will decide what the next steps are. So if you have a BRCA1 or a BRCA2 mutation, your world narrows in to that world of how you can prevent getting cancer. So there's a lot of early screening, maybe earlier screening than usual for things like mammograms and maybe transvaginal ultrasounds, and the whole clinical world descends on you here. And especially with something like cancer, you might even consider prophylactic surgery before onset of any disease. So, so it's not easy to make it's one thing to make a product that is of high value to everybody and that people can get the most out of. It's a completely different thing to make it popular. So getting scale for a product like this required for us to build the earlier product, right? We had to do something that first familiarized people with their genome, was a softer intervention, something they could change their diet on, something that they could just change the type of fitness activity that they conducted, and then introduce this to them, because then they were almost like they were ready to take, okay, what next is on the table. And we found that by combining these two as a strategy, we find that the actual access and reach to the entire population is significantly improved. It helps us that various organizations, including I think one of your sponsors, Biopic, works on creating health consciousness in communities, and we're working with them very closely with them to deliver these types of tests to their clients. What I see today in terms of wanting to be healthy, people wanting to be healthy, the numbers of people, young people who want to stay healthy and ahead of disease is far higher than in my time. We kind of dealt with things when they happened to us. And now I see people actually preemptively trying to hold off on these things from happening. So I'll stop here just because in the interest of time, and I know that possibly coffee is the next stop, and probably everybody needs a bit. But I'm happy to stop here and sort of take any questions that you may have, any curiosities that you may have to discuss. But I would like for you to take away is genomics for everybody. Yes, it is. At what level do you want to interact with genomics? Thank you. Sir, please. So, yeah. So NutriDNA alone retails in the market, including a nutritionist counseling session, for 9,999 rupees. That's it. 9,999.
That's not much.
Sir, we can take your sample soon.
Yeah, sure.
People come to your house. Someone will come to your house and take it, yeah. This whole thing is now on your phone. You can just book it. Someone comes to your house, takes it. You get your report on your phone. When you deal in the consumer market today, there's nothing that's difficult.
That's right.
So, yeah.
That's nice. Yeah.
Sir, I actually wanted to bring that up. I had a completely different presentation today from this. And in that presentation, I wanted to say that 20 years ago, this would have cost us 50,000 rupees to do this exact same test. Right? The cost of sequencing has come down so low, and I was part of that journey with Illumina to bring this cost low. It's the least of the cost that we worry about now. It's the tube collection companies that probably make more than the sequencing folks do, right?
Yeah, Ashok.
Hi.
Ashok, as a scientist, okay, the question is, for the future that we see, right, and for the generations that's going to come, let's say that genomics is going to be the way of life. It's going to be more reliable, more sensitive. What is that one test which you think would be just welcome at face value? Like, no convincing, no hardship, like it's just going to be on the counter.
I would stop one level before that, Madhavan. If we could convince people to eat well and exercise right, we don't even have to do any testing. right? Half the problem is that memo has been lost on the way, right? And that's why companies like us enjoy the fruits of that kind of non-compliance, right? So, but if you were to ask me, I'd say a basic test like NutriDNA would be a good starting point to get going.
Thank you.
Yeah, sure.
So I just have a question here. So when you're talking about some of these tests and the reports, is there a factor that you take into account? Because like you said, the reports show up on the phone.
Yes.
Now, different human beings process data differently.
Yes.
They're the emotional people, they're the logical people practically. Do you ever think of having an emotional quotient kind of thing built up so that even reports are personalized from individuals to individuals?
Wonderful question. Thanks for asking. I did mention, I'll repeat it again, that there is a nutritionist session built into this. So that is our first human contact that we make with someone. And at that point of time is when, trust me, more than just what you need to eat comes out, right? I mean, there's a lot of agony aunt things that happen on such calls because that is the nature of any counseling session. And counselors are trained, in fact, to deal with that kind of questioning. So we bring that in at the very end.
I mean, it's fantastic because as you see you're using generative transformers here, it would just take enough data to understand how to probably even build counseling sessions.
Yes.
Once you have enough sample space going ahead.
I can tell you that the question is very valid, and we tried and insist that we have a human contact somewhere in the entire channel of things that we do. But the real quality interaction is at the level of the counselor.
Hi, Dr. Ashok. So what do you think,
Yes.
sorry, what do you think is the biggest barrier to entry in consumer genomics? Say,
price. I mean, Dr. Murthy is kind. He says 9,999 is fine, right? But the number of people who say, oh, I thought it's 99, then I'll buy it. You'll be surprised. See, two things are happening simultaneously. Cost of sequencing is coming down. People's ability to afford or disposable income is going up. These two things are happening simultaneously. At some point of time, they'll match. That resonance will happen. And these things everybody will do.
are we able to connect with the niche market, say, who can afford it? Are we able to connect and communicate better with them?
That is one of the advantages in general. The association of being affluent monetarily also means you come with an education and things like that. You'll be surprised that how many people who have an education don't really want to go into the details of what this has. Just tell me the solution, right? And which is where the session with the nutritionist, or maybe even just from the report itself, if they have a clear instruction, okay, from now on, eat spinach and don't eat burgers, for some reason that makes them feel like they've got their money's worth. In the second test that I described, Genomic Health Insights, you'll be surprised. There's a large group of people, because close to 80% of the people don't get any call-outs, any disease call-outs, okay, for cancer or anything like that. The folks who pay 12,000 rupees for that test and don't get a call-out have actually complained to me, "Itna paisa de diya hai hum, kuch nahi mila humko." Right? So consumers you need to communicate effectively and differently with them. And I won't say we've cracked it, but we're learning.
Hi Ashok, Shubhanji this side. So so NutriDNA, wonderful product. So when we come to the Genomics Health Insights.
Yes.
So just out of curiosity, we tried something very similar back in 2018.
Yes.
So we did a lot of corporate engagements, a lot of reach out, similar hall packed with people, full of excitement. But at the end of the day, the conversion rate was less than 1%. Nobody was really interested, and they were like, "Jab hoga dekhenge," you know.
We beat you by 0.2%. We're at 1%.
Why no now? You know, when we face something, we'll see.
Yeah.
So that's one part. Second part is that, once somebody finds some risk, some probabilistic risk.
Yeah.
As you said, there is nothing that we can offer as a solution because you don't have it yet.
You can. So maybe I misspoke if that's what you heard. So what we do have is some kind of path forward. Okay, now that you are, let's say, BRCA, you're talking about genomic health insights, am I
Yes,
correct?
yes,
Yeah. Now that you are BRCA1 positive, we put you onto a network of doctors, clinicians who are actually educated on this. Now, you know, as I do, that not all clinicians know associations of genetic mutations with outcomes, right? So we put them in the right space. The second thing is, we have a genetic counseling session built into what we do, where we lay out a floor plan of what their next steps should be. Now, we don't actually walk them through those next steps, and we don't see them through to the end of their journey, but we have to let go at some point of time because it is their personal journey to take forward.
So, like for example, we had intense discussion with medoncs and oncologists and clinicians regarding this. One of the problem that we faced was that, say you gave an example of BRCA1, BRCA2, and as a follow-up, one can go for a mammogram or routine tests. But that is again exposing the individual to higher radiation risks.
Yes.
When you don't have it already.
Yes.
Secondly, the routine, somatic markers or the panels that we recommend patients.
Yes.
Once you have it,
Yes.
these are usually recommended based on other outcomes. It can be, once you do, some kind of diagnostics with the biopsies, kind of, cell-based studies and so on, and then you recommend those tests. Only once you have it, once you have some indications from the biopsies and so on. They are ideally not recommended when you don't have it. There is no biopsy information, there is nothing. So in those cases, have you come across any recommendation from the clinicians that yes, there are still some tests or something which is not as hazardous, that can be done on a routine basis to keep track of things?
So there is an entire product that I didn't talk about, and there are about 5 slides beyond this, something called Cancer Spot that Strand has come up with. It's a liquid biopsy based approach to understanding whether you are currently harboring cancer or not. The formula is differential methylation of cancer cells and their extrusion into the serum plasma, and the assessment of any aberrant signal. If there is one, then you're probably harboring a cancer. And that is a non-invasive, minimally invasive, right? Because you do have to draw blood, but it's a minimally invasive way in which you take this forward. So as part of that ecosystem, we'd like to, once all the thorough evaluations, clinical evaluations on the product are complete, we'd like to have that as a next ecosystem product in, let's say you do have a predisposition, then here is a non-radiological way in which one can actually assess the next steps.
Thank you.
Thank you. Thanks for your question.
Just one last question following up. When you said what doesn't work, the bottleneck is the price. I probably have a different approach. I mean, do you think the problem is how we deliver? Is it the delivery vehicle that probably is not attractive to customers? I mean, today Apple is saying,
we're experimenting.
I have an FDA, you can do blood pressure monitoring with a watch. Now, is there a way we can create reports which are interactive, which are not on the phone or wearables or something?
See, we are in that, the client we talk about today is in that diversity that we can never please everybody all the time. We're going to get only some people interested in what we do and how we do it. And we got to hope that in time, those who are not interested develop an interest. So we just got to wait, right? That is the yeoman's version of laboriously working through the market. But we really appreciate it when organizations, other organizations like Biopeak et al., raise the health conscience of folks in general, and then more people are coming to us, asking us, hey, tell us more about these tests, right? I don't think in the 1980s you could have said HbA1c and it would have been a common parlance. Today everybody knows the number, forget what HbA1c is, right? These things will percolate into communities. It's a question of how sticky we can make it and how useful we can make it in them, impacting their lives. So the onus on us to prove. That's it? Yeah, thank you very much.
Thank you, Doctor, for that insightful talk where access expands, impact implies. I would like to call upon Dr. Raj Vagre for felicitation of our speaker. The agenda says as we come to the close of BioVerse 2026, it is only fitting that we hear from one of the scientific pillars behind this journey. I'd like to invite Benna for the closing note.
Yeah, very good evening all. I'm usually not very comfortable with a mic, and today I'm all the more emotional and a bit nervous. So if I go wrong somewhere, I'll have a disclaimer in the beginning itself. Pardon me for that. And if I miss mentioning anybody, it was not intentional. I'm not going to say a very formal thank you note by mentioning names today. But what I want to say here is each one of you in this hall today, and many who couldn't make it, have been very critical in bringing us to where we are. So the event itself is a thank you note for all of us who have been with us. So 10 years ago, I think we started Azooka blindly, especially me, because I was a hardcore academician, no clue about business. I didn't know how to build products. So I blindly jumped into this. But we started with a question. Can we build molecular biology grade products from India for the world? Can we make our labs safe? So that was the question we started with. But at that point in time, I think all of you would agree that most of the science which backed healthcare systems was concentrated only in one part of the world, and India was known only to adopt it. So we dreamt of changing that, not by importing, not by assembling, but by building it ourselves. But little did we know the challenges that we are going to face through this journey, right? So initially, if I have to recollect, a lot of people used to mock us. More than me, I think Alex had to go through a lot of people who mocked, why don't you do just software? Why do you want to get into a space you don't know? There were a lot of rejections, and because this was a domain, unlike software, which needed patience, which needed us to build IPs, which needed perseverance and precision. So it was a real challenge at that point in time, and little did we know that the ecosystem, or at that point in time, building deep science innovation from India was going to be this big a challenge. So despite or amongst all of this rejections, I think I'm standing here today primarily because what I realize in hindsight is we were still not alone. And I think that's what made Azooka what we are today. So our friends and family became investors and collaborators. They trusted in us when we had nothing. I still remember Vardarajan transferring a good amount of money when we didn't even have a bank account officially. So I think we've had friends who have really supported us, many such friends who really supported us through this journey. Our teams, I can go on talking about our teams. We've been fortunate enough to have wonderful teams throughout this 10 year of journey. Our teams became family. And I think I have to give credit to my entire team who has pulled up this show without me or Alex stepping in. I've been on a 1 month vacation to help my son with his boards. Alex was down with fever, and it is only them who have pulled this up. Without, I would say mine and Alex's contribution in this is 0%. So everything goes to the team. So they became our family. Every team member of Azooka, we've had these kind of experiences. I can share as many anecdotes as possible. And for us, family also became team. I remember during COVID times, we used to work with volunteers. We had to ship at least 20 to 25,000 units in a day. We were just beginning to start manufacturing, this was kind of challenge, and we could find only few people to work with us. At that point in time, I remember my kids, I remember Brinda, her children, we used to take tubes home and they used to label. They used to do the labeling. So that's the kind of support that we've received. So where, everybody came together and saw us as family and helped us in whatever possible way they could. And the next I have to say our mentors. I think they became our pillars and our compass. They went beyond advice. I think the support that we've received from them is much, much more than any advisor would play. I think I mentioned how Murthy opened the doors for me, which changed my entire life. Professor Savitri, Tintorong was fully developed in her lab for 5 years. She gave me a fellowship to build that. She gave me her lab. Like she said, she gave a team for me to work with and even chemicals. During tough times, she used to ask me to order chemicals from her lab once. And many such instances. We've had advisors. He specifically doesn't want me to mention his name, but we've had advisors who gifted us money when we were very low. Not a small amount, but a substantial amount. So they've gone beyond just playing the role of advisors. They've done things what as parents, as some of our parents don't. So now I really realize why Guru comes before God. So this is coming from my heart. Sorry for getting emotional here. So that's the kind of support we've got from our mentors. And ecosystem partners, I think when we started, the ecosystem was not as great as it is today. But still, we could materialize or we could survive for 6-7 years only with grant money from DBT, DST, and these sources. We've raised more money from grants than from investors. I think the ecosystem is also maturing now, primarily to support the kind of work that we do. And I've also been associated with some associations like Catalyst for Women Entrepreneurs, Sonda Connect, who helped in small ways, but that was very significant at that point in time. And I definitely want to mention here that many a times our vendors have silently supported us because we've defaulted on payments. And if I have to say, sometimes I think I cleared a bill that was 5-year-old to our patent attorney just last year, and she's been very supportive and patient with me. So our vendors have been silent supporters, and I think we don't underestimate the support that they've given us. And finally, our customers, right? I think Kumar, I would say Kumar was our first customer who trusted in our products, replacing imported German products. He was willing to try, he was willing to switch, he was willing to move. And after which, a lot of other customers followed, trusting that India can innovate and build these kind of products. So I think our customers became our proof. So we are what we are today, primarily because of all of you. So I just want to leave with one note. Although we started with redefining healthcare and redefining diagnostics, it was not easy. And although the journey has been challenging and very stressful, at the same time, very rewarding because we could see our products touch real lives during the pandemic. So once we started working during the pandemic, it opened us doors to understand that sample collection itself was a problem with the Indian healthcare system. So that was a realization which helped us build a platform which enables biological sample collection. You can say from your eye tear to your poop samples. We have a range of products where sample integrity, nucleic acid integrity is maintained from your eye to your blood, tissues, poop, urine, and whatnot. So that's what we have successfully built. So I'm proud that we are the only company in the world who has a solution for all the different types of biological samples, while all the companies or all our competitors, German or Canadian or US competitors, have 1 or 2 products on those lines. And Tintorung is the only safest molecule that binds to DNA, which has been reported in the past 6 decades. So I think that is what we have done. And although we say that the Indian healthcare system is fragmented and we have to catch up a lot, but I think today's talks clearly demonstrate that all the speakers have demonstrated that we are redefining our healthcare system with microbiome science, with consumer genomics, with brain mapping, with smart stethoscopes, and I don't know what more that we are not aware of. So I think we are already playing this part together. So I'm proud of building and solving these problems from here, for here, and with you all. Thank you. Thank you for being with us. And although it's a 10-year journey, I think this is going to be the beginning for us, and we need your support and guidance for the next 10 years. Thank you all for coming and making the event a grand success. Thank you.
Thank you, Dr. Bena, and to all of our speakers, partners, and attendees for making BioVerse 2026 truly meaningful. Azooka turns 10 today, but as Dr. Bena said, this gathering makes one thing clear: the next decade is just the beginning. Please join us for Thai tea. Have a wonderful evening, and thank you.
There was an idea. Reimagining molecular biology, engineered. There was an idea. Reimagining molecular biology, engineered for safety. Elevating Indian healthcare to global genomic standards.
Redefining what's possible, where innovation meets safety.
कोरोना वैश्विक महामारी के खिलाफ
Most countries did not have the infrastructure to handle the cases.
We did something called as an RNA wrapper, which is alternative for viral transport medium.
At Azooka Labs, we protect the truth inside every sample. May it be blood, saliva, tissue, any sample, we stabilize it. We envision a truly decentralized home-to-lab diagnostic ecosystem, where samples are collected at home and clinically validated insights are delivered anywhere without ever stepping into a clinic. It's time to move beyond traditional blood tests, toward layered health intelligence, where microbiome, hormones, and wearable data provide a complete health overview, turning health data into an asset. Azooka Labs.
There was an idea.
Reimagining molecular biology, engineered for safety.
Elevating Indian healthcare to global genomic standards. Redefining what's possible, where innovation meets safety.
कोरोना वैश्विक महामारी के खिलाफ
Most countries did not have the infrastructure to handle the cases.
We did something called as an RNA wrapper, which is alternative for viral transport medium.
At Azooka Labs, we protect the truth inside every sample. May it be blood, saliva, tissue, any sample, we stabilize it. We envision a truly decentralized home-to-lab diagnostic ecosystem, where samples are collected at home and clinically validated insights are delivered anywhere without ever stepping into a clinic. It's time to move beyond traditional blood tests, toward layered health intelligence, where microbiome, hormones, and wearable data provide a complete health overview, turning health data into an asset. Azooka Labs.
There was an idea.
Reimagining molecular biology, engineered for safety.
Elevating Indian healthcare to global genomic standards. Redefining what's possible, where innovation meets safety.
कोरोना वैश्विक महामारी के खिलाफ
Most countries did not have the infrastructure to handle the cases.
We did something called as an RNA wrapper, which is alternative for viral transport medium.
Azooka Labs, we protect the truth inside every sample. May it be blood, saliva, tissue, any sample, we stabilize it. We envision a truly decentralized home-to-lab diagnostic ecosystem, where samples are collected at home and clinically validated insights are delivered anywhere without ever stepping into a clinic. It's time to move beyond traditional blood tests toward layered health intelligence, where microbiome, hormones, and wearable data provide a complete health overview, turning health data into an asset. Azooka Labs.
There was an idea.
Reimagining molecular biology, engineered for safety.
Elevating Indian healthcare to global genomic standards. Redefining what's possible, where innovation meets safety.
कोरोना वैश्विक महामारी के खिलाफ
Most countries did not have the infrastructure to handle the cases.
We did something called as an RNA wrapper, which is alternative for viral transport media.
At Azooka Labs, we protect the truth inside every sample. May it be blood, saliva, tissue, any sample, we stabilize it.
Marra. Wait.